all joining us virtually. Hearty welcome to all of you. On behalf of ICAR Central Marine Fisheries Research Institute and our co-organizers, Swadeshi Science Movement Kerala, SSMK, Vigyan Bharati, Vibha, and Amrita Vishwa Vidyapeetham, it is my distinct honor to welcome all of you to this national seminar and brainstorming session on the Murburn Concept Discussion Forum. Science is inherently non-dogmatic. It thrives on empirical rigor, critical inquiry, and a relentless commitment to truth. Instead, sometimes it's not so, but indeed, evidence acts as the Supreme Court in scientific inquiry. There is nothing. Evidence is the mastermind or the dictator in science. And empirical data accumulates conventional wisdom, often gives way to deeper, more nuanced paradigms. We have witnessed this profound shift before in molecular biology. For decades, the classical consensus held that only protein coding genes were useful, relegating nearly 98% of the human genome to the bin of junk DNA. Today, modern genomics has shattered that dogma. We now know that non-coding sequences play vital roles, acting as master regulators of gene expression, harboring crucial microRNA loci, and maintaining genomic stability. A similar conceptual revolution is unfolding in bioenergetics with the Murburn concept. For years, reactive oxygen species ROS were viewed almost exclusively as toxic, metabolic byproducts responsible for cellular degradation and pathological states. However, pioneering evidence now suggests that transient diffusible reactive species, DRS, are not mere agents of chaos. Instead, they serve as obligatory constructive role in core cellular physiology, mediating electron transfer, powering ATP synthesis during oxidative phosphorylation, and maintaining systemic cellular hemostasis. To debate, dissect, and advance these novel research frameworks, we are privileged to have a stellar panel of leaders and scientific mind with us today. I extend a warm welcome to Dr. K. Muralidharan, the Program Coordinator, SSMK, who will enlighten us about the scope of this program. Hearty welcome to you, sir, to this function. A special welcome to our international speakers joining us online, Dr. George from Medical University Varna, Bulgaria, and Dr. Laurent from Antwerp, Belgium, for their upcoming theme addresses. We are honored by the presence of our guests of honors, Shri P.A. Vivekananda Pai, Secretary General Vibha. He is an engineer by profession, a close friend. I welcome him heartily to this function. And today we have with us Professor Dr. D. Vasudevan, Dean of Medical Research, Amrita University. This particular concept is going to have maximum impact in the medical research. So his presence adds value to this program. Welcome you, sir. My sincere greetings to Professor V. Shivakumar, President of SSMK, who will deliver the presidential address. Welcome, sir. This is his second visit to CMFRI. Welcome to you, sir. There is a very special welcome to our Chief Guest, Professor P. Raveendran, Honorable Vice Chancellor of Calicut University, who will deliver the inaugural address. He is a man of chemistry. He will know the cellular physiology much better than any of us. And there is no better person than Professor Dr. P. Raveendran to be the Chief Guest of this function. Hearty welcome to you, sir, for this function. And the entire discussion of this program happened because of Dr. Murali Manoj. He is a founder, conceptual leader of Murburn Concept. This program is entirely because of Sri Murali Manoj. And from CMFRI side, this has been Dr. Ashish Khandelwal who has been coordinating this program. This potted plans is a count of numbers also, so that I will not miss anyone on the stage at least. And finally, it's Dr. Ashish Khandelwal who coordinated this program with the entire team of partners. In addition to what we are going to discuss today and dissect today, we have two major activities happening as part of this inaugural session, which I have not informed the chief guest. The first thing is a memorandum of understanding signing of CMFRI with the Environmental Defense Fund India. Environmental Defense Fund India, they promote different conservation activities what are happening in the country. And recently, CMFRI has seriously embarked on environmental approach to fisheries management. Fisheries management itself is a very difficult concept in the sense like the temperate science. They always try to say that what they are doing is right and rest is wrong. But in India, we have developed a methodology called stratified multi-stage random sampling method. Unlike the Western concept, this was vetted by Food and Agriculture Organization of United Nations, and we have been following it. And this has become the backbone for any tropical multi-species fishery what is happening in the country. So we'll be having an MOU signing with EDIF. Mr. Mondal and Mr. Vijay, both of them have joined from the EDIF side. They will be, I welcome them here. They will be signing MOU with us. The second activity is on the publication of the code list of the major fishes. Indian tropical marine fisheries is having around 2,500 species of marine fishes and related species, out of which in the harvest database, around 1,200 species are there. Out of 2,500, 1,200 is coming to our harvestable database. That indicates that we are using only 50% of biodiversity in the usual way when we are catching the fish. So this is done with the methodology I was mentioning earlier, and we have a strong database under National Marine Fisheries Data Center, which is at the first floor. If the dignitaries find time, I will take you there and show like how we are doing it. So we will be publish. Our team is here. It was led by Dr. J. Shankar and his team. So we'll be doing that code list publication also today. I also extend my greetings to my colleague, Dr. Ashish Khandelwal, Senior Scientist at CMFRI, along with all researchers, students, and participants who are attending this forum. Science progresses when we dare to question established frameworks with the fresh evidence. I am confident that over the next 2 days, this forum will serve as a vibrant crucible for critical debates, scientific clarity, and path-breaking ideas. So for this to happen, you are the people who have come here. You can clap for yourself for joining along with us in deliberating, discussing, and understanding this Murburn concept. Welcome to all the delegates who are sitting here. There are other organizers of all these collaborating and hosting institutes. I welcome each and every member who belongs to ICAR-CMFRI, VIBA, SSMK, and Amrita, who has joined today with us. Hearty welcome to you. This is one of the premier institutes in the country, which is going to celebrate its 80th birthday. It is as old as independent India. And this institute has been taking the pioneering research and related efforts with respect to the marine fishermen who are involved not only in the fishing activity, but protecting the coastal heritage of this country. Since they are there, many of the attacks to the coast is prevented because they serve as foot soldiers in the sea. So it is this institute who is doing the marine fisheries census also for them. We go to every fisherman household. There are 1 million fishermen household. Either me or my representative go to every fisherman household and do the census for the government of India. There are a lot of other initiatives. If you are interested in marine fisheries, mariculture, a glimpse was shown in the video. You can go to our social media handles, like it, follow it, and you can understand more about marine fisheries and mariculture activities. This is a wonderful field. There are many youngsters who are joining us for doing research, for being part of our research. So welcome all of you to this wonderful institute. If there are issues, you can come to 5th floor. I sit at 5th floor. It is in the northern extreme of 5th floor. Complaints you can tell to me. Good things you can tell to others. So be brand ambassadors of CMFRI as you walk out of this esteemed institute. So let us join together in taking science forward. Thank you. Jai Kisan, Jai Anusandhan, Jai Hind.
Thank you, Dr. Grinson, for your warm and gracious welcome. Setting the tone for the day of scientific dialogue, collaboration and innovation. ഊഷ്മളമായ സ്വാഗതം ഓരോ മഹത്തായ സംഗമത്തിന്റെയും മുഖമുദ്രയാണ്. നന്ദി. Now it's about the program. Every successful scientific event is guided by a clear vision and a well-defined purpose. To briefly introduce today's program and its objectives, may I respectfully invite Dr. K. Muralidharan, the distinguished scientist, the visionary science communicator, and the program coordinator of Swadeshi Science Movement Kerala. കാര്യപരിപാടിയുടെ ആമുഖവും ലക്ഷ്യവും അവതരിപ്പിക്കുന്നതിനായി സ്വദേശി ശാസ്ത്ര പ്രസ്ഥാനത്തിന്റെ പ്രഗത്ഭനും ദീർഘദർശിയുമായ പരിപാടി ഏകോപകനും ഡോക്ടർ കെ മുരളീധരൻ സാറിനെ ആദരപൂർവ്വം വേദിയിലേക്ക് ക്ഷണിക്കുന്നു.
നമസ്തേ. ജഗദ്ഗുരു അമൃതാനന്ദമയി നമഃ. Respected scientists assembled here, all of you are scientists. So it is a very proud moment for Swadeshi Science Movement to interact with the young and experienced scientists in under one roof. And this has happened only because of the wholehearted support, unconditional support from Dr. Grinson George, director of this institute. Otherwise, also in many Swadeshi Science Movement activities, he help us, he support us. So on behalf of the Swadeshi Science Movement Kerala, the state unit of Vigyan Bharati, the largest assembly of scientists in the world, I welcome you to this event. When I want to tell you about this event, I thought I will start with a story, rather a piece of history. It was in 1922, one gentleman called Chandrasekhar Venkataraman was in his voyage to Europe, and he was mesmerized with the deep blue of Mediterranean Sea. And he also looked up the sky. Suddenly it flashed in his mind, it is not just a reflection of sky. That was the explanation given by one of the very eminent Nobel laureate, Lord Rayleigh, on this aspect, because he only explained why the sky is blue through the scattering of light. Coming back to India at Kolkata, Raman continued his work in the Indian Society for Cultivation of Science. Incidentally, that organization is celebrating its 150th birthday anniversary this year, and it was founded by Dr. Mahendralal Sarkar, 29th July 1876. So he continued his research, and in 1928 February, 20th February, he announced the Raman effect that all of you know. Immediately he published and also booked a ticket to receive the Nobel Prize in the next year. That is 1929. Unfortunately, he didn't get any announcement. Again, he was so sure he will get it, and 1930 he booked the ticket, and during that period, that year, it was announced. So why I mentioned this story to you is, we have to see the three important qualities as a scientist. One is courage. It is not that any scientist, whether it is Nobel laureate or anybody, simply tell something, simply you read black and white, it is not the end of science, or it is not, may not be the fact. So you should have the courage to question if you feel something is wrong. Then you should be convincing to yourself. That conviction is also very important. Then immediately you have to communicate. See, Raman immediately communicated his findings in the Indian Journal of Physics. He didn't wait for sending it abroad or anything. So these are all very important. And when you see the work of Professor Kailath Murali Manoj, some of these qualities you can see. It was, we were in association with him, that is Swadeshi Science Movement Kerala, associated with him during his presentation at National Institute of Technology, Calicut, during the 30th Swadeshi Science Congress. Thereafterwards, we were, some of my members always interact with him. And he is a proponent of the concept that is the mured burning or Murburn. So we assembled here to discuss that. And it explained what is the importance of oxygen in our life. Everybody know, without oxygen we cannot live. Not only we, any living organism. But what exactly the role? Till he proceeded, the general feeling is that oxygen is to receive electron and to convert it into water at the end. And many Nobel laureates endorsed that view. Many textbooks were taught us, many theories built upon that. But when he looked into it, he found that the role of oxygen is not that low. It is there for production of large number of reduced oxygen, SRDs. Then, and that large number of ROS make the condition for many cell activities, suitable conditions for good or bad cell activities. And this is a path-breaking finding. Based on this idea, many advancements took place in different disciplines of science, which we will be discussing in this 2 days seminar. And it will be first he will be giving a keynote address, and there will be discussions joining online as well as present here also. And I request all of you to interact, debate on this issue critically. You need not be mercy to anyone. You can criticize, you can ask questions. Then only the science will progress. So I wish a very learned experience to all of you during these 2 days. And it should strengthen our understanding about the universe without hurting anybody. Om Sahanavavadu, Sahano Bunaktu, Sahavidium Karivavai, Tejasinaadimastu, Tejasinaadimamastu, Navidyashi. Om Shan, Shan, Shan.
Thank you, Murali sir. That is a lucid and insightful overview, clearly outlining the vision and scientific significance of the unique discussion forum. Leksham Vyaktamayal Yatra Kudadal Arthavathavunu. Nandi sir. Now it's time for the theme addresses online. The first one is from Bulgaria and second from Belgium. Yeah, the professor. Okay, the first professor from Bulgaria, Dr. George Chaldeko, is 87 years old and has some connectivity issues. I think his views will be presented along with the second theme address. That's to be presented by Dr. Lauren Jacob of the Karel D. Groot University of Applied Sciences and Arts, Antwerp, Belgium. He's not available. Okay, we'll have it afterwards, after some time, when the connectivity issues are getting resolved. Okay, it's about 6:30 a.m. there, so we'll go for the addresses by the guest of honor. Science attains its highest purpose when knowledge reaches society and transforms lives. We now move to the addresses by the distinguished guest of honor. We are indeed delighted to have us with us. Our first guest of honor, a visionary who has consistently worked towards fostering scientific temper and national development. Through the promotion of science and innovation, may I respectfully invite Dr. P.A. Vivekananda Pai, Secretary General, WIPA, to address this August gathering. Aditi Sandesham നൽകുന്നതിനായി വിജ്ഞാൻ ഭാരതിയുടെ സെക്രട്ടറി ജനറൽ വിവേകാനന്ദ പൈ സാറിനെ ആദരപൂർവ്വം വേദിയിലേക്ക് ക്ഷണിക്കുന്നു.
Thank you. I was very keenly listening to Muralidharan ji because I have been hearing about this concept. Abgaji has been mentioning, Muralidharan ji has been mentioning about Murburn concept. But I am an architect and I am waiting to understand more about it. Dr. Grinson George, Director CMFRI, who has been a pillar of support for all our activities. Respected Dr. D. Vasudevan, Dr. Kumar, President, Swadeshi Science Movement Kerala, respected Dr. P. Raveendran, Ashish Khandelwal, K. Muralidharan ji, Murali Manoj ji, all the dignitary students present here in this very important seminar. Why I say this is important? Just yesterday, we had a discussion at Bhopal. So someone was describing about the evolution of many of the scientific concepts. He mentioned one incident, which we all know. Galileo had a public experiment. He dropped a 1-pound ball and a 10-pound ball from the top of the Pisa Tower. The whole city had assembled to see, because the understanding till then, or the theory till then, was that the lighter weight will fall slower and the heavier weight will fall faster. Galileo said it is not like that. It is the gravity that is acting on everything in the same way. He was not accepted. So he made this public experiment. And to the surprise of all who had assembled there, both of them came down together at the same speed. So Galileo proved himself right. But more interesting was the reaction. Because those who followed the other theory, they propagated that Galileo was capable of black magic. Dr. Grinson was talking about scientific enquiry. And when we look at the story of scientific enquiry, this resistance has always been there. Another example, we all know, just now Muralidharan ji talked about Chandrasekhara Venkatraman. Another great scientific genius from our country was Jagadish Chandra Basu. We all know how he worked on microwave receiver, how his research predated Marconi's and all that. But then he moved on to physiology, plant physiology especially. And he was looking at how the plants and metals were responding to various stimuli. And he came out with his theory. He came out with the crescograph and all that. But how that theory was looked upon in the initial stage, there is a wonderful book by Kunal Ghosh. He's from IIT Kanpur, I guess, called Unsung Genius, A Life of Jagadish Chandra Bose. A part of it is technical, but otherwise also I think every science student, if possible, should read that book. Amazing how a scientist has evolved. So in J.C. Bose's case, he tried to propagate this idea of how plants also have life and even metals respond. He made a presentation at Royal Society also. And then there is this mention in this book. I will just read that. Sir John Burdon Sanderson specialized in the study of muscles and nerves, particularly the movements of the Venus flytrap. It was Charles Darwin, famous for the theory of evolution, who had drawn Sanderson's attention to the electrical physiology of the Venus flytrap, a plant that traps a fly or an insect using slow, deceptive movements. So Sanderson was the acknowledged authority on electrical physiology, and all looked to him to open the discussion after the paper, after J.C. Bose presented his paper. And he began by paying compliments to Bose's physical work, then continued. It is a great pity, Sanderson's reply. It is a great pity that Professor Bose should leave his own subject for other fields that properly belong to the physiologists. That is the first comment. that this is none of your business. Professor Bose's paper is still under consideration for publication. He may consider altering the title. Then he actually suggests to Professor Bose, see, this is not your field, but I suggest the electric response to certain physical reactions. The term response belongs to the physiologists and not the physicists. Further, regarding the electrical response of ordinary plants demonstrated by Bose, I am of the firm opinion that it is absolutely impossible, since I myself have tried to detect it for many years and never found any. It simply cannot be. And who is saying this? One of the greatest scientists of that time. But this is just one incident. Then this book continues to show how Bose was hounded after his return to India and at Presidency College also for the mistake that he proposed this theory. But today we know Bose was right. So any new theory always has its own questions and it should raise questions. But there has been very difficult experiences also, whether in the case of Galileo or Bose, and if we look at the different experiences of various scientists and technocrats, it has always been there. But let me compliment Murli Manojji as well as all the organizations, Amrita, CMFRI, SSM Kerala, and all others, and all who have joined here, that let us try to explore this new thought. Because even science otherwise also is now at a crossroads. In the case of consciousness and matter, in the case of matter and energy, in the case of when we talk about quantum mechanics, there is no definitiveness today, which we already had long back in the times of the Newton. So that is where we are, and a lot of discussion is happening. I am very glad, and all best wishes to this new concept and the discussions upon that. Thank you. Namaste.
Thank you very much, sir, for your inspiring address, which just beautifully reminded us that the true strength of science is enriching society and improving human life. ശാസ്ത്രത്തിന്റെ യഥാർത്ഥ മഹത്വം സമൂഹത്തിന്റെ നന്മയിലേക്കുള്ള അതിന്റെ സംഭാവനയിലാണ്. നന്ദി, സർ. The next guest of honor is another magnificent personality. Experience is a silent architect of wisdom, and wisdom lights a path for future generations. It's now a great honor to invite a distinguished medical scientist, an eminent academician, and an inspiring teacher, whose contributions have profoundly influenced medical education research in our country. May I respectfully invite Professor Dr. D. Vasudevan, Dean of the Medical Research, Amrita Institute of Medical Sciences of Amrita Vishwa Vidyapeetham, to address the gathering. Amrita Vishwa Vidyapeethamile, Amrita Institute of Medical Sciences Medical Research Dean, Professor Dr. Damodaran Vasudevan Sarne, സദസ്സിനെ അഭിസംബോധന ചെയ്തതിനായി ആദരപൂർവ്വം ക്ഷണിച്ചുകൊള്ളുന്നു.
Respected Vice Chancellor of Calicut University, Professor P. Raveendran, Dr. Grinson George, Dr. Muralidharan, Professor Murali Manoj, Shri Vivekananda Pai, Professor Siva Kumar, and Dr. Ashish Khandelwal, and my dear colleagues. Today we have assembled here to look into a new concept of Murburn concept that is promulgated by Professor Murali. The science, as usually, will progress something like that. There will be one concept, and then there can be an alternate concept will come forward by somebody or some other group, and there will be profound discussion on pros and cons of various concepts, and then the better things will be taken. Sometimes both will be absorbed together or symbiosed together. These are the way by which the science grow. As such, today we know that when we take the foodstuffs, that is broken down in the body, and finally all these minute particles will be oxidized. And this oxidation, during this oxidation, the energy is released, and that energy is trapped by chemical method by ATP, and that much everybody agrees. However, the general concept, present day general concept is that the final oxidation inside the cell is taking place inside the mitochondria, and in the mitochondria, the foodstuffs are made into smaller particles, and that is made into NAD or NADP, NADH, and this NADH is oxidized by a series of electron transport, and this is called electron transport chain. And during this process, hydrogen ions are produced. Hydrogen ions are transported across a membrane, and along with that, there will be a hydrogen ion concentration on the other side. And that hydrogen ions will come down through a narrow pore. And during that process, the ATP is generated. That is the present day accepted concept. 3 Nobel Prizes are already given to different persons at different time intervals. This what I have told will take about 50 or 60 years of research work, and that ultimately led to 3 Nobel Prizes. Therefore, this is the accepted knowledge. Here is a new concept, the Murburn concept, and we are here to critically analyze that and define the novel research frameworks. That is absolutely necessary, because there are, in my mind, there are 3 objections, or not objections, there are 3 difficulties to accept by all persons by the new concept. One is that, we know that electron transport chain is present. The proteins are there, that is, everything is present inside the mitochondria. And what is its work? If that is not, the electron transport chain is not necessary, then what is its work? Therefore, we have to identify, of course, it is also sometimes producing ATP, and how much ATP is produced. The second question is that how much ATP is produced through the old concept, and how much is produced by the new concept. That is the second question that we have to answer. The third concept that we have to answer is that, the Murburn concept that is taking place, the free radicals are generated everywhere inside the cell, and that free radicals are generating by oxidation, generating the energy. In that case, how it is regulated? These 3 major questions have to be answered, and that for that new research has to be initiated. And this is the framework that we are here. For this purpose, this is to generate the critical debate we have to generate here, and we are all, and the best wishes for Professor Murali. First, we have to congratulate to bringing a new concept, and then I wish all the best for that. The general community, general scientific community, I will accept that, provided we give all the answers. Thank you.
Thank you very much, sir. Your scholarly reflections, enriched by decades of academic experience and scientific leadership, have truly elevated and inspired this inaugural session. Anubhava Sampathinte Prakasham, Talamuragale Vazhi Nadutthunnu. Nandi, sir. It's time for the presidential address. Leadership is not measured by the position one holds, but the vision one inspires. Ladies and gentlemen, Every scientific moment gains strength through visionary leadership. It's now our proud privilege to invite our distinguished leader and visionary advocate of science and innovation, Professor Dr. V. Siva Kumar, President of SSMK, to deliver the Presidential Address. And he is a Professor of the Department of Health Sciences Research, Amrita Vishwa of Medical Sciences Kochi. ശാസ്ത്രബോധത്തിന്റെ ദീപശിഖ ഉയർത്തിപ്പിടിക്കുന്ന സ്വദേശി ശാസ്ത്ര പ്രസ്ഥാനത്തിന്റെ പ്രസിഡന്റ് പ്രൊഫസർ ശിവകുമാർ വേണുഗോപാൽ സാറിനെ അധ്യക്ഷ പ്രസംഗത്തിനായി സാദരം വേദിയിലേക്ക് ക്ഷണിക്കുന്നു.
Thank you, Dr. Vivian. Namaste. Good morning to one and all. On the dais, we have some of the stalwarts sitting here. Professor Dr. D.M. Vasudevan, who is an authority on biochemistry. Professor Raveendran, sir. And of course, from Swadeshi Science Vigyan Bharati, we have Dr. Shri Vivekananda Pai ji. And I should also thank Dr. Grinson George for actively encouraging scientific discussions and granting the permission to Swadeshi Science and Vigyan Bharati and sharing their infrastructure and encouraging us in all the scientific debates. So I must congratulate Murali sir for this program, because when he initiated this talk that Swadeshi Science has to take the lead in organizing this program, I recall the initial discussion with Dr. Murali, and I was so happy that he is a faculty of Amrita Vishwa Vidyapeetham. So I felt proud that such a stalwart pioneer in the fundamental research is part of my university. I recall when in the year 1992, I was doing my post-graduation in Madras University, and there was an invitation to listen to the lecture of James Watson. James Watson, the founder of the DNA double helical proponents, and my professor, my head of the department, P. Nandakumar of Madras University, he told that, okay, we have got this invitation, we have to go and participate in that lecture. So we all went to Anna University, and it was a very big, so many students were there from across Madras at that point of time, and we could listen to him. And my professor was an authority on genetics, Professor P. Nandakumar. Very strict and tall guy, more into cricket than into genetics. But I recall his lecture. He used to talk about genes, chromosomes, and he used to talk about junk DNA. Junk DNA means the vast majority of the DNA in our cells, they don't code for anything else. They are just there. And they only a fraction of the DNA actually translate, and all of the vast majority of them are silent, means they don't do anything visible. So they were termed junk DNA. This was in the textbook. I recall him talking about junk DNA in the class. Now, subsequently, we understood there is nothing like junk. They are all silent. They are all having genetic information, which in turn provides signals later on, even though they don't visibly act on in any of the transcription or translation. But those junk DNA have silent information which directs. So there is nothing like junk. So science has evolved I would say science has matured to come to terms with the reality. So I would say Murburn concept is also sounds to me like that. So it's an evolution of the human thought itself. I recall Professor Sultan Ismail, my sir, who is the first DSc from Madras University, used to give me a very beautiful example. He's an authority on ecology. He used to say, as a young child, he used to ride a bicycle. And I'm also a very avid cyclist as a schoolboy. So what he used to do, he used to say, his hobby was to dismantle the cycle parts. Now, dismantle during the holidays, he used to dismantle the tires, wheels, the chain, everything, and then assemble all together. That was his favorite hobby. I've also done it. This was his favorite hobby. And these days, one day he put everything together. And after that, the cycle looked very normal. And when he looked out, there was one nut and one bolt missing. And he doesn't know where to fit this. The cycle looks very normal and ready to ride. But this nut, he doesn't know where exactly he has removed from the cycle. But we don't know where to put it. But he said, I get to know it when I start riding the bicycle. When I started riding the bicycle, oh, some noise is coming. Oh, this nut belonged to the bell or to that pre-wheel or somewhere. So, ladies and gentlemen, the message of the story is nothing in nature is a waste. That is the message he told me. That he used this example. Professor Sultan Ismail, he's still there. He's still very active. He is the first DSC of Madras University, and my teacher is still. I'm in touch with him, and the fundamentals of molecular biology, which he taught me, helped me so much when I got into industry. That what used to take 8 years, I got promotion in 2 years because the fundamentals were so strong, thanks to my teachers. So I recall when I heard about this Murburn concept, I recall my junk DNA lecture of my teacher, who later on himself corrected that science is evolving. And also my teachers, the cycle example, that nothing in nature is based. We grow in wisdom to understand. And as Vivekananda Pai ji gave the example of Galileo and how he was, finally, when the evidence was shown, people finally want to put him aside by saying that he's capable of black magic. To that extent, people can go to, not to give up on their prejudices. In fact, I just completed one course on Indian philosophy where we talk about consciousness, the mind-matter relationship. And the first chapter I kept was the title of the paper was by Dr. Dean Radin. His beautiful paper, the title of the paper is called Pathological Skepticism. If you go to the Google and find out Dr. Dean Radin, the title of the paper is Pathological Skepticism. Beautiful paper, just 2 or 3, I think not a paper, it's an introduction to a journal, something like that. And Dean Radin is an authority on science and consciousness studies with more than 500 papers. Like he's among my friend circle. And I made his paper because the skepticism is so intense that they don't want to give up their positions. Questioning their concept is questioning themselves. They are so attached to it. So there needs to be a evolution in that, not only in the scientific realm, but also in the spiritual dimension of the scientific community also. Like the whole world, what we see is changing, right? But there is an unchanging entity, and that is consciousness. So the science is moving towards that dimension, what our ancients had explained so many years, so many millennia of years ago. So I wish this conference all the best. I look forward to listen to the deliberations of this conference. And on behalf of Vigyan Bharati and Swadeshi Science Movement, I thank CMFRI for granting us this facility. And also thanks to Professor Muralidharan for initiating this discussion. And that's how science moves forward. One day I recall telling Vasudevan sir a famous quote about, I didn't know who told it, science moves from funeral to funeral. Sir, you may recall this. Science moves from, not by step, funeral to funeral. And sir told me it was by Maxwell. So what does it mean? People are unwilling to, give up their, I would say, not differentiating between science and prejudice. So it's very important. As I want to quote here the quote from Dr. Speth from Harvard. He says, I thought all the great problems in the world like pollution poverty can be overcome with 30 years of science. With 30 years of science, all problems in the world can be avoided. But I am wrong, because what is needed is, what is the problem is apathy, greed, selfishness, and prejudice. This is, if you go to the Google, Dean of Yale, Dr. Speck, S-P-E-T-H. This is the quote, beautiful quote. I thought 30 years of science will overcome all the problems of the world, but no. The problems of the world are caused by greed, apathy, selfishness, and pettiness. And for that, we need to have a spiritual revolution. I'm not saying this is from the Yale University Dean. So with that quote, let me wish all the best for this program, and I look forward to listen to the deliberations. Thank you very much.
Thank you very much, Shivakumar sir. Your visionary presidential address has inspired us to pursue the scientific excellence with renewed commitment, intellectual openness, and collaborative spirit. ദൂരക്കാഴ്ചയുള്ള നേതൃത്വം പുതിയ സാധ്യതകളിലേക്കുള്ള കവാടങ്ങൾ തുറക്കുന്നു. നന്ദി. Now, may I request Dr. Kailath Murali Manoj, the promulgator of Murburn Concept, to deliver the felicitation address as a guest of honor, please. A couple of words from you, please.
I thought it opportune. Thank you. I thought it opportune to address the gathering here when the stalwarts are here. We know now the cosmos that we see is vast nothingness, spread, entirety. In there, there's a little mass, very little mass. In those mass that accumulate, many gigantic stars. There's a small planet, Earth. In that, we know there are living beings. Among the big living beings, blue whales, powerful ones like tigers, we humans, we look for answers. So that rational, critical, curious temper that we have, tiger, one-to-one, we can't face a tiger. It's much stronger, faster. But then humans, tiger hunts the same way as it did 10,000 years back, 100,000 years back. We humans, we are different. Why? Because we have society, we have culture, we believe in this discourse. People gather information, we share to the forthcoming generations. This is core. That is possible because of family. And now institutions, 3 institutions have lent their strength. And I want to take this opportunity to thank these 3 institutions. First and foremost, Swadeshi Science Movement Kerala and Vigyan Bharati, the largest scientific movement in our country. I wanted to thank Menon sir, Namburi sir, Abgaji, Selvaraj, and our Paiji, Venugopal sir, for lending the core support. Then I wanted to thank our CMFRI director, Dr. Grinson sir, who without much hesitation at the Muralidharan sir, sorry, I missed out his name. Sorry, the key, that's a problem. Muralidharan sir of SSMK. Then our Grinson sir, he agreed, and then Ashish Khandelwal ji, he was running around for the last 1 month. Thank you. And my own institution and our Vasudevan sir, part of that institution, but he put Murburn as a component of the textbook. 2 people who could not make it here. Beyond Amrita, there is our Bipin Nair sir, registrar, he is on the line. I wanted to thank also 2 people, very instrumental in our having this meeting today. One is Dr. George Chaldakov. He was supposed to address. Yesterday night, he said he couldn't make it. He had sent a message So that message was supposed to be relayed through Dr. Laurent Jakin from Belgium. That didn't happen. Hopefully he'll join sometime and he will give the messages. And most importantly, Professor Raveendran from Calicut University, my longtime friend from IIT Madras, and Professor Venugopal, he has come with him. He's also here. So Professor Raveendran braved the very unfavorable weather, drove all this. Why should people who don't get any money, any financial benefit out of this support it when it is going against the establishment? We need better answers. And that's the reason. Thank you, sirs. Professor Ravi's rational temperament, we know while we were studying in IIT Madras. I don't know how many of you will remember Ramar Petrol. It was about to be launched. He had opposed it and he had to face a lot many threats for that. So he also invited me to Calicut University. And first time, in their chemistry conference they have occasionally. So he had invited me to talk. Because of these great well-wishers and curious minds, that's how science advances. And you people, young generation who have come to listen to what Murburn concept is, it's about completely rebuilding the way we think about life. Life is what affords thoughts. Thoughts sustain life. So this is about that. How do we think? How do we move? Why do we breathe? How does plant tap sunlight? All that is possible because of institutions and individuals. My utmost gratitude and respect to the people who bring that. This thing can work only if there is gratitude and respect. And my gratitude and respect to the people on the dais, to the institutions, and to all of you who have come here. Thank you so much. Thank you so much.
Thank you, Dr. Kailath Murali Manoj sir, the pioneer of Murburn concept, and for the enlightening address. Now comes a felicitation and release ceremony. Ladies and gentlemen, every scientific gathering leaves behind more than memories. It leaves behind knowledge. It leaves behind partnerships and lasting commitments. Now we arrive at a very special segment of today's inaugural session, the felicitation ceremony, followed by the release of publication, exchange of MOU, and the product release. First is the felicitation of Dr. Kailath Murali Manoj. Yeah. Great scientific ideas are born when curiosity meets courage. Today, we honor a visionary scientist whose pioneering thought has given birth to the Murburn concept. It's our proud privilege to felicitate Dr. Kailath Murali Manoj, the promulgator of Murburn concept, for his remarkable contribution to scientific innovation. May I request Dr. Vivian Namburi, the curtain core of the Swadeshi Science Movement, and the eminent professor of photonics in the International School of Kochi University of Science and Technology, CUSAT, to facilitate here. Felicitation is done with a shawl as well as a memento. Dr. Kailath Murali Manoj Sarne, Swadeshi Science Movement in the Peril, Elia, Snehovaram Samanichukundu, Ponade Anichukundu, Adarikinai, CUSAT, photonics professor, Dr. Vivian Namburi Sarne, Vinay Purvam, Vedi Likshanikinu, Nani sir. There is a citation, I will read it out first. SSMK expresses its deep sense of pride and honor to celebrate our warm association with Professor Kailath Murali Manoj, affectionately known as KMM. Professor Manoj's seminal work in introducing the Murburn concept represents a monumental paradigm shift in biological science, fundamentally redefining how oxygen sustains life at the cellular level. His pioneering insights have challenged and rewritten the established paradigms of bioenergetics originally conceptualized by past Nobel laureates. SSMK warmly felicitates Professor KMM in recognition of his relentless efforts to place Bharat's scientific achievements at the forefront of modern research while honoring and connecting them to our ancient scientific heritage. A big round of applause, ladies and gentlemen. Thank you. Next is a release of the publication. Every documented species is a chapter of story of our oceans. Every scientific record strengthens the foundation of sustainable fisheries. And now we have the privilege of witnessing the release of the publication. Taxonomy-based caught list of marine resources encountered in Indian fisheries. A significant contribution by the Fishery Resource Assessment, Economics and Extension Division of ICAR-CMFRI, authored by Dr. J. Jaishankar and team. Others are also requested to be on dais. The team consists of Sindhu K. Augustine, Sijo Paul, Deshma A.R., Manjish R., Manu V.K., Dr. Somi Kuriakos, Dr. Mini K.J., and Dr. Ildo Varghese, and Dr. Vinay Kumar Vase. Whoever are present here as authors may please be on the dais. Release of the one of the finest publications. May I request the dignitaries to kindly release this valuable publication. Dr. J. Jaishankar is absent. The team consists of Sindhu, Sijo, Reshma, Manjish, Manu, Dr. Somi, Dr. Mini, Dr. Eldo, Dr. Vinay. ശാസ്ത്രീയ രേഖകളാണ് സുസ്ഥിര മത്സ്യവിഭവ പരിപാലനത്തിന്റെ അടിത്തറ. അറിവിനെ രേഖപ്പെടുത്തുമ്പോഴാണ് അത് തലമുറകളുടെ സമ്പത്താവുന്നത്. നന്ദി സർ. അപ്ലോസ് പ്ലീസ്. Thank you. Now it is time for the MOU. Together, the science and stewardship can create a sustainable future. Collaboration is a key that transforms shared visions into meaningful action. We now move to the significant moment of collaboration, the exchange of MOU, Memorandum of Understanding between ICAR-CMFRI and the Environmental Defense India Foundation, EDIF, making a shared commitment towards strengthening environmental conservation and sustainable initiatives. May I respectfully invite Sri Hisham Mandal, Honorable Director of Environmental Defense India Foundation, EDIF, to kindly join us on stage for this important occasion. Please. ശാസ്ത്രവും സംരക്ഷണവും കൈകോർക്കുന്ന ഈ ധാരണാപത്ര കൈമാറ്റം സുസ്ഥിര ഭാവിയിലേക്കുള്ള ഒരു സുപ്രധാന നാഴികക്കല്ലാവട്ടെ. A big round of applause, friends. Sri Hisham Mandal, Director of EDIF, would like to present a publication entitled Sea Change, authored by James Walkman and Amanda Leland, to the Director of CMFRI. May I request Sri Hisham Mandal to present the publication to the Director as a token of love and appreciation. For this exchange of Memorandum of Understanding between EDIF and ICAR-CMFRI. ശാസ്ത്രവും സംരക്ഷണവും കൈകോർക്കുന്ന അമൂല്യ നിമിഷം. ഈ ധാരണാപത്ര കൈമാറ്റം സുസ്ഥിര ഭാവിയിലേക്കുള്ള ഒരു സുപ്രധാന നാഴികക്കല്ലാവട്ടെ. Thank you, sirs. Now it's time for the product release. From the depths of the ocean to the touch of everyday life, marine science continues to inspire innovations that benefit humanity. We now move to the special moment, the release of Kadalmeen seaweed soap, an ocean-inspired innovation enriched with natural fragrance, developed through the expertise of marine biotechnology. This dermatological tested product with reduced microbial reduction potential represents the successful translation of marine research into a value-added wellness product. This innovative creation has been developed by Dr. Ashish Khandelwal of Marine Biotechnology, Fish Nutrition Health Division of ICAR-CMFRI. May I once again respectfully invite the dignitaries on the dais for the release of the Kadalmeen seaweed soap, a beautiful fusion of the ocean essence and scientific innovation. കടലിന്റെ സമ്പത്തും ശാസ്ത്രത്തിന്റെ സർഗാത്മകതയും കൈകോർക്കുന്ന നിമിഷം സമുദ്ര വിഭവങ്ങളുടെ സാധ്യതകളെ ജനോപകാരപ്രദമായ നവീനതയാക്കി മാറ്റുന്ന സംരംഭം. Let's give a resounding applause, friends. Thank you. respected dignitaries, these symbolic moments represent much more than ceremonies. They reflect our shared commitment to collaborate, knowledge disseminate, and then scientific progress. May these partnerships continue to inspire meaningful research and innovation in the years ahead. സഹകരണത്തിന്റെ കൈകോർക്കലാണ് ശാസ്ത്രത്തിന്റെ മുന്നേറ്റത്തിന് ഏറ്റവും വലിയ ശക്തി. നന്ദി. Now it's time for the major event of the function, inaugural address. Ladies and gentlemen, every scientific conference has its defining moment, a moment that symbolizes the formal beginning of a journey of ideas. Universities are not merely the centers of learning. They are the birthplaces of ideas that shape the future. വിജ്ഞാനത്തിന്റെ വെളിച്ചമാണ് സമൂഹത്തിന്റെ വഴികാട്ടി. Today, we have arrived at the very moment. It's now our singular privilege and honor to invite the most distinguished guest, an eminent academician, a visionary educationist, and a respected institution builder, Professor P. Raveendran, Honorable Vice Chancellor, University of Calicut, to deliver the inaugural address. വിജ്ഞാന നേതൃത്വത്തിന്റെ പ്രഭാവവുമായി കാലിക്കറ്റ് സർവകലാശാലയുടെ ബഹുമാനനായ വൈസ് ചാൻസലർ പ്രൊഫസർ പി രവീന്ദ്രൻ സാറിനെ ഉദ്ഘാടന പ്രഭാഷണം നിർവഹിക്കാൻ ആദരപൂർവ്വം വേദിയിലേക്ക് ക്ഷണിക്കുന്നു.
The respected director of CMFRI, Dr. Grinson George, Sri Vivekananda Pai, Secretary General Vibha, Professor Vasudevan, Dean Medical Research, Amrita University, Professor V. Kumar, President, SSMK, Professor Ashish, Dr. K. Muralidharan, and other dignitaries on and off the dais. Good morning to all of you. I'm extremely happy to be here, to be part of this national seminar and brainstorming on the Murburn concept. Particularly, we have Dr. Manoj, who is here on the stage, who brought out this concept of Murburn to explain many of the physiological and other problems which we have been discussing for a long, long time. And my hearty congratulations also to CMFRI today for the kind of translations which they have been doing with respect to the research they are doing, as evidenced by the product release which we had and the MOU and other things. Well, I think Murali invited me as a friend to this program because he was my junior, and not in my research group, but in another research group in the biotechnology department at IIT Madras, and we are together in the hostel. He came a couple of years later than me. He was there. In the later sense, I always used to say that Murali was abnormal even in those times. And there is, I don't know whether you are familiar that there was a Limca, they had an ad in those times that being, it is boring to be normal or something of that sort. So we always used to say that, Murali, he actually imbibed that with him. I didn't see him for a long time after that. And later when I saw him, he talked to me about his Murburn concept. When he told me Murburn, then I asked, Mur is for Murali, and burn, that is Murali burning. Is that? So he said the story is almost the same. Like Murali has been burning for quite some time now with his Murburn concept to get accepted. And he has been publishing a large number of papers in high impact journals. And of course, he had an equal or more number of rejections of his concept from several areas. So he was, in fact, he was quite disappointed with these rejections. So I told him, like, if you don't have to be rejected, because if your concept has to be accepted, more than 100 high impact publications or thousands of publications will have to be buried before they accept your Murburn concept. And many of these things, like the Professor Vasudevan, in fact, wonderfully introduced the topic of Murburn, or at least what ETC, electron transfer chains, how the present system of explanations work. And what Murali has come up with, the new concept which he has come up with. See, one of the interesting and important aspect of science is that when you actually propose a new concept, particularly when you have a concept which has been awarded, some theories which has been awarded several Nobel Prizes. And if Murali's Murburn concept gets accepted, I think it is, I was listening to Professor Charlako the other day. It is worth several Nobel Prizes, not one. But that exactly is the challenge. That exactly is the challenge. When somebody comes and proposes a new concept, it is not very easy that it gets immediately accepted. It is also not necessary that it should get accepted. This is something which we all have to have in mind. Because when new scientific theories are proposed, we should have an open mind. We should not be in a hurry. I know Murali is in a hurry, but it is not always true that this works in that pace, because you have so much of literature available based on the ETC. And if you have to get it accepted, all these have to go for further evaluations. Most of these concepts will have to be evaluated, re-evaluated, reassessed in terms of the Murburn concept, and to see which one is advantageous to explain the observed experimental data, because I think Murburn has again issues more number of experimental data. I am sure that that will come out in terms of the theory that he has proposed. So it's very important that we have to wait. We have to wait. We have to have this, open mind. We have to have a balance. And I'm sure that eventually, when a new concept has been put forth, this is very important. What I liked about is Murali's thing, he has been incessantly pushing it forward. He has something new to tell the world, and he has been trying to put these things together to explain many aspects of human physiology and many aspects of the bioenergetics in terms of the Murburn concept. Although I wish, his theory gets immediately accepted, but I know that that is not the way that science works. It takes time for people to check and have the checks and balances in place and to explain which one prevails. So I wish Murali all the best, my friend, good friend, Murali, all the best. And I congratulate him for getting such a wonderful opportunity to have a brainstorming session on Murburn, because rarely we actually have seminars being conducted like this, being conducted based on a topic, a new topic that being presented by somebody. Now, unfortunately, I am not an expert in this area. So, although Dr. Grinson George said that being a chemist, it's not purely a chemistry problem alone. So, I'm not an expert in the area, but I have been listening to Murali, and I found that there are several things. For example, the cyanide poisoning issues, which I have been talking about. Why such a small amount of cyanide is enough for somebody to die? Why there are no stoichiometric issues that are not explained, when the present cyanide poisoning mechanisms are explained. So there are lots of such issues worth really brainstorming. We need to actually think all these things afresh in terms of the Murburn concept. And I wish Dr. Murali Manoj all the best. And I'm sure that the theory will get accepted over a period of time. And there will be significant scientific advancements based on this. Because when some new theory comes, it is always like this. Even if the theory fails sometimes, lot of scientific discussions and new discoveries will come with that. I still remember one thing that probably, I do not know whether you are, the Miller's theory of the prebiotic soup, with the experiment which he did in Chicago University with urine. And that was one theory about how life could have evolved in reducing environments. And in electric shock, he actually put all these gases inside a small glass vessel and produced an electric spark. And you could produce all those biomolecules which we think are probably the molecules of life. So he actually proposed a theory, went very well. In fact, initially it was rejected, it got accepted, but later, after long time, people said the environment was not a reducing environment. But the kind of scientific discussions that Miller's theory brought in was enormous. So this is how science works. We need to really work on these, and it has to be subjected to detailed discussions, experimentations, and over a period of time. I'm sure that the Murburn concept, I wish all the best to Murali, that it will get accepted and he will get the recognitions that it needs. Congratulations again, and I formally inaugurate this program. Thank you very much.
Thank you so much, sir. Your inspiring inaugural address has reaffirmed the vital role of higher education in nurturing innovation, inspiring young minds, and strengthening the culture of scientific excellence. വിജ്ഞാനത്തിന്റെ വെളിച്ചം തെളിയുമ്പോൾ നവോത്ഥാനത്തിന്റെ വഴികൾ തുറക്കപ്പെടുന്നു. ചിന്തകൾക്ക് ദിശയും ഗവേഷണത്തിന് പ്രചോദനവും നൽകിയ ഈ ഉദ്ഘാടന പ്രഭാഷണത്തിന് ഹൃദയം നിറഞ്ഞ നന്ദി, സർ. What about the pending theme addresses? Dr. Lauren Jacken from Belgium is available online. Okay. No, vote of thanks. Gratitude is the most graceful conclusion to every meaningful beginning. കൃതജ്ഞതയാണ് സംസ്കാരത്തിന്റെ ഏറ്റവും മനോഹരമായ ഭാഷ. As we come to the close of this inaugural session, it's now our privilege to invite Dr. Ashish Khandelwal, an accomplished marine biotechnologist, the emerging innovative researcher, and senior scientist ICAR-CMFRI, to propose the formal vote of thanks. Dr. Ashish, please.
Respected chief guest, dignitaries on the dais and of the dais, dear students, members of the media, ladies and gentlemen, a very good morning to everyone. It is indeed my privilege and honor to propose the vote of thanks on behalf of the ICAR, Central Marine Fisheries Research Institute Kochi, Swadeshi Science Movement Kerala, Amrita Vidyapeetham, Viva, during inaugural sessions of this national seminar and brainstorming session on Murburn Concept Discussion Forum. At the outset, I express our heartfelt gratitude to our chief guest, Professor P. Raveendran, Honorable Vice Chancellor, University of Calicut, for graciously inaugurating this seminar and for delivering an inspiring inaugural address. Your words have motivated all of us to strengthen interdisciplinary scientific research and foster meaningful collaborations. We convey our sincere gratitude to Professor V. Shiva Kumar, President of the Swadeshi Science Movement Kerala, for his visionary leadership and constant encouragement and inspiring presidential address. Your efforts in promoting scientific awareness and indigenous scientific thoughts have been truly recommendable. Our sincere thanks are due to our distinguished guest of honor, Sri P. A. Vivekananda Pai, Secretary General Viva, and Professor D. Vasudevan, Dean Medical Research, Amrita, for sharing their valuable insights and enriching today's program with their thought-provoking address. However, Dr. George Cheldakov, he did not attend the program, but he has sent his message for warm for this particular program for this particular program to a great success. I extend my sincere gratitude to Dr. Grinson George, our beloved director, ICAR-CMFRI, for his warm welcome, encouragement, and unwavering support in hosting this national seminar. His commitment to promoting interdisciplinary research and scientific excellence has been instrumental in making this event as a success. I also express my heartfelt appreciations to Dr. K. Muralidharan sir, Program Coordinator, also Dr. Kelat Mulli Manoj sir, from Swadeshi Science Movement Kerala, for his meticulous planning, dedicated coordinations, and tireless efforts in organizing this program so efficiently. Today's seminar will demonstrate that scientific progress is driven by curiosity, evidence, open discussion, and constructive debate. The stimulating presentations and brainstorming session will generate new ideas and open promising avenues for future interdisciplinary research on the Murburn concept. I sincerely thank all the distinguished scientists, medical experts, academicians, researchers, faculty members, and students for their enthusiastic participations and valuable contributions. I also extend my gratitude to the Swadeshi Science Movement Kerala, Viva, Amrita Vishwa Vidyapeetham, and all collaborating organizations and institutions for their wholehearted support in organizing this national scientific event. Finally, I express my sincere gratitude to every participant who traveled from different parts of the country and abroad through online mode to join us. Your presence reflects a shared commitment to scientific excellence, collaborative research, and the pursuit of knowledge. Thank you, everyone. Jai Hind.
Thank you, Dr. Ashish. Your heartfelt expression of gratitude has beautifully acknowledged the collective efforts that made this inaugural session a memorable one. Indeed, every successful event is built upon the silent dedication of many hands and many hearts. നന്ദി പറയുന്നത് ഒരു ഔപചാരികത മാത്രമല്ല, അത് നമ്മുടെ സംസ്കാരത്തിന്റെ മനോഹരമായ അടയാളമാണ്. Distinguished dignitaries, the respected delegates, ladies and gentlemen, with this, the inaugural session of the discussion forum on Murburn Concept formally comes to a close. We thank everybody towards the success of this inaugural session and sincerely hope that the scientific deliberations ahead will stimulate meaningful discussions, encourage collaborative research, and inspire new dimensions of scientific discovery. Before I conclude, allow me to leave you with one thought. Science is not merely the pursuit of answers, it's a courage to ask questions that no one has dared to ask before. May these 2 days discussions kindle that courage. May these 2 days conversations inspire tomorrow's discoveries. And may this gathering strengthen our collective commitment to advancing science for the benefit of humanity. അറിവ് പങ്കുവച്ചാൽ വളരും, ചിന്തകൾ കൈകോർത്താൽ കണ്ടെത്തലുകൾ ജനിക്കും. ശാസ്ത്രം മനുഷ്യരാശിയുടെ നന്മക്കായി മാത്രമെന്ന് മുന്നേറട്ടെ. Thank you so much. Have a wonderful and intellectually enriching conference. After the inaugural session, all esteemed dignitaries may kindly assemble here for a group photograph. Now, with the hearts united in pride, united in purpose, may I request everyone to kindly rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
Thanks a lot, ladies and gentlemen. Now, all are cordially invited to the stage for a group photograph. The first session will commence after 10 minutes. There's a break.
The ones who have joined online,
The
please,
interested
there's a break.
participants can come to the
10
dais
minutes.
to have a group
Dear
photograph.
Marko, Mahendra, Afsal, Sukumar sir, Babu, Tofia, there'll be a 10-minute break. I request you to join, please, after. There's a 10-minute break.
Sure, no problem.
Then there's my talk. Please don't go away, those who are online.
Professor Kalath Murali Manoj. We have few discussions joined by online as well as presented here also. So it is my pleasure and privilege to welcome and introduce our panelists and the speaker. Already we have told most of the things about Professor KMM. Right now he is a faculty at the Ettimada campus of Amrita Vishwa Vidyapeetham. Then this session is chaired by Professor P. Raveendran, Vice Chancellor, Calicut University. Then along with Professor P. Venugopal, again a chemistry professor from Punjab University as well as now at Calicut University. Then Professor Dr. Damodaran Vasudevan, again he was there in the inaugural session, Dean Medical Research, Amrita Vishwa Vidyapeetham. And Dr. Grinson George, Director, ICAR, Central Machine Fisheries Research Institute. I request all the dignitaries to occupy their seats. Let them sit. As a token of love and affection to all these dignitaries, we will honor them with a bouquet of, a flower check. Load my slides, please. First slide.
Pointer where now? All right. Thank you. Good morning. Oh, sorry, it's almost afternoon now. Dear chairs and students, scientists, passionate scientific ideas pursuers, I'm here to present my ideas on Murburn concept. This talk is about the broad outlines of claims, terms, context, and impact. In fact, you can rephrase the title, How Do We Live and Why Do We Die? I'm a professor at Amrita Vishwa Vidyapeetham. With my thanks already mentioned in this talk, I will not be mentioning thanks. So let's get on with it. I started these Murburn presentations first with the Parent Institute of Science in India at IISc. And that slide and poster was made by Vivian, who is sitting there, challenging Peter Mitchell's theory. And then we conducted 2-day workshop at IIT Mumbai. And then in 2024, we had a 3-day symposium at Amrita. This is the next in the series. It's a 2-day discussion forum. This discussion forum was the brainchild of actually our Muralidharan sir, sitting in the front there. I believe that science should be discussed and debated, not just one person comes, speaks, and just walks away. The format is all of you sitting there. If you ask good questions, you get prizes. And there is also going to be a quizzes for the students at the end. All right. So please pay attention. Before I proceed, our Kumar sir also already mentioned some of the ideas that I have already got to say. Now, scientific temperament, all of the professors and teachers and seasoned professionals talked about the critical spirit that science is pursued. Science is just one of the facets of human endeavor. We, as humans, we believe in loyalty, art. There are so many other codes of pursuing science. But when we have science, the basic idea is if you try to, the purpose of science is to explain reality or natural phenomena. So imagine you have a statement made that an ant goes in a circular path. You take points and you try to plot them. And if it agrees with the circle's equation, you say that it's okay. It abates by that theory. But then you can have points here, 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 and all that. And yet, it might not be close to reality. There could be a few points, and it need not be a continuous phenomena also. But you can still fit the process to a circle, disregarding some aspects of reality. You have to explain, the theory should explain all aspects. And when you come to the math, if these are the experimental points, you can fit the mathematical fit with different types of equations. They all might give statistical analysis, correct? And what happens is usually when a theory has a limited scope, let's say it can explain only this volume, what we do is insert a cone into that, and all incompatible observations or findings, you try to put in that. So basically, this is got to do with how science is pursued. And when we try to explain this image by Escher's waterfall is very, very important. Explanation is about, not about math, not about equations. It's how we try to understand something. So if you see this image, you'll see from the second floor, water falls down, it turns the turbine, and then it pushes the water all the way. At any particular point, if you see, the artist has made it impossible for us to discern. But from our natural experience, we know that this is highly unlikely. It is not possible. So we have to understand that when we have to say that something is right, we need infinite points to say something is right. But to say something is wrong, we need only 1 point. It doesn't abide by the circle. So in science, validation or ratification is secondary. In science, critical, questioning, and refutation or falsification is primary. So the scientific philosopher Karl Popper said, scientific advancement fundamentally rests on the premise practice of empirical falsification. So falsification, refutation is key. So in science, claims, evidence, arguments are important. Of course, consensus is also important, but that is a secondary aspect. It can come later. All right. So this is the philosophy of science that I shall pursue. And Professor Shivakumar already said advances one funeral at a time by Max Planck. And it's very interesting to note that Max Planck opposed, in quantum electrodynamics and other quantum physicists that came, these people opposed, even Einstein opposed. But later on, they became accepted. So here is one thing that I want you to think about, St. Georgi. Dionysians and Apollonians, both are interesting and important. Dionysians, like me, they make radical changes, but then systematic improvement and expansion of known fields, that's also important. So these are the fundamental aspects of science that need to be considered, and the role of critics in the advancement of science. So, like I said, claims, evidence, argument. So first I stake my claims. The claims are Murburn models established continuum of physics in biology. Physics is the mother of all sciences. Math is the language of it. And in Murburn models, completely revamped the most fundamental reaction on planet Earth, that is photosynthesis, which gives us oxygen that we breathe and the food that we eat. Then each cell goes through respiration. That process also, then how body produces heat, how you see me, how you process information, how intelligence, sensing, you name it, Murburn is involved. Metabolism, drug metabolism, epigenetics, post-translational modifications, pharmacokinetics, pharmacodynamics, ecology, mechanophysiology, how bacteria move, toxicity of cyanide, immunology, origin, sustenance, termination, evolution of life. What Professor Raveendran was talking about, Urey-Miller experiment. This is more aligned with that. So fundamentally, what I claim is the most fundamental aspect of life is impossible to be explained without invoking Murburn concept. That's a tall claim to make, I know fully well. But I hope at the end of these 2 days, or at least this talk, you will see some substance in it. So what I say is, we know that living beings, the reality has got, we try to understand reality by physics, and that has got basic foundations in information, then space-time and quantum theorizations. And fundamentally, life we try to study here at this level, the world of objects, biology, chemistry, and chemical physics. In this, I shall argue that Murburn concept bridges the the domain of non-living. From physics, life emerges through Murburn. So Murburn concept, central dogma, that is DNA gives RNA, RNA gives proteins, proteins catalyze all that. Then cell theory, cells are the fundamental units of life. So the organization of life, if you need to understand how powering, coherence, homeostasis, electromechanical activities, sensing response, without invoking Murburn, you cannot. This is a fundamental claim of my talk. So before I go into the details, I would like to tell you, each talk in this meeting would be focusing on, first, what are the classical views? What are the things that you learn in textbooks? Then, what is wrong with those ideas, briefly? And then, what is the Murburn model? And then, comparison. That's how we are going to do it. It's up to you to take it or leave it. All right? So, the classical is cells are the fundamental units of life. I'll quickly go over this. DNA codes for proteins, and proteins have beautifully shaped active sites which can bind to various molecules, ions, all other kinds of components that are involved in cells. So the selectivity and specificity of reactions are governed by the genes that code for the proteins which have a particular active site, and that can distinguish. I know Professor Raveendran looks different from Dr. Grinson or Professor Vasudevan, because I know their topology has registered the space. I can map it, I differentiate. Similarly, at a molecular level, the catalysts, those agents that transform reactions, they fundamentally identify molecules with this mechanism of topology. So this lock and key active site or induced Wick theory of identifying is a core aspect of control in biological reactions. Professor Vasudevan asked, how do you control in Murburn? So we'll come to that. So one other aspect of the classical theory is very highly deterministic membrane pumps and channels. The cell functions through generating electric gradients or chemical gradients, and they are harnessed by some kind of molecular level intelligence, and pan multiphasic orchestration is also sought by localized determinants. So this is what it is, whether it is enzyme substrate reaction, electron donor acceptor, or receptor ligand. So serial, sequential, and multi-phase spatial coordination requires this theory, okay? So next aspect is powering, the most fundamental aspect. Now, we know a phone or this projector, it has some printed electronic circuits, and you apply a potential, which is a driving force. Now, our body is a soft matter, fluidic. We don't have possibilities for wiring like we have in these systems. So, but the classical system, whether they are mitochondria are the powering units of the cell, they say that like what Professor Vasudevan sir already mentioned, NADH, after food is broken down, ultimately goes through glycolysis, Krebs cycle, and then which makes NADH. Then NADH, through the electron transport chain, the protons are pumped out, creating a gradient. For this idea, Peter Mitchell got the Nobel. And then the gradient is dissipated through complex 5 or ATP synthase. And this idea got rotary ATP synthesis. It goes round and round to churn out ATP, just like our mills we have. So these are all Nobel recognized ideas. And then these are the fundamental ideas for physiology advocated by Cambridge School of Thought, the British ones, and taken over by the rest of the world. That is, a cell's membrane is kind of a sentient sentinel. That is, it is some kind of a smart guardian which can identify all molecules and ions. It can estimate them and pump in a particular stoichiometry using the energy currency that is produced in the powering module. So cellular powering, coherence, homeostasis. Coherence is how does, whether a cell is unicellular or multicellular, how does it behave as one unit? Homeostasis, how does it maintain its integrity? I have eaten something, my cells should retain the same composition after 2 days. If I eat 2 aspirin tablets and it doesn't go out of my body, then I will cease to be Murli. I'll be Murli plus aspirin, right? So that is not acceptable. So all this is classically central dogma. That is, genes give proteins, proteins have active sites. They control everything through active site chemistry. That's the classical theory. Now let's come to the limitations of the classical theory. What do you think are the simplest things that even a 10th or 12th class students can understand? Fundamentally, a mitochondrion, which is considered the powerhouse of this cell, it was given a Nobel Prize. It has got very low volume and the H plus concentration is very low. And if you multiply it with the Avogadro number, you should get the proton number. So practically, mitochondria or cyanobacteria, they are practically aprotic. There is no proton to be pumped. And if you imagine proton can be formed from water, this is a highly endergonic process. So ATP synthesis, the ATP bond itself is only 36 kilojoules per mole. Water is even stronger. So there is no way Peter Mitchell's proton pumping can work because mitochondria is so small that protons cannot be available freely. This is 10th class chemistry. So when I sent this to the Nobel laureate who is still alive in UK, he said, yes, you have done something. I cannot refute it, but let the world decide. So this is first point. Second point is, we know basically that when a membrane is based in a particular way, the potential, positive, negative charge like that, you cannot have ions going crisscross at their own fancy. There should be some rationale. And also in cyanobacteria, you see, it can carry out both photosynthesis and respiration. And one requires water breakdown to form the nicotinamide reduced agent. And in the other, it requires nicotinamide reduced agent to form water. And in both processes, ATP should be formed. And in these, the electron transport chain, the idea that Sir was talking about, it requires some common elements. How will cytochrome C know that I have to do photosynthesis now, I have to do respiration now? It is not possible by this serial kind of idea. And most importantly, how can things avoid this inevitable chemistry where oxygen can easily gain an electron to make superoxide? It's highly exergonic, very facile. So oxygen would shunt every step. It is not possible to get that wiring. So fixed TM polarity, electrons and protons cannot go in an unrealistic deterministic mandate is yet another fundamental argument. Third argument, Stephen Hawking made one very beautiful example in Brief History of Time. If you take a hot cup of tea and you drop it, the tea cup breaks, the heat dissipates, the water spreads. You cannot have the same process that is highly spontaneous. You cannot have it reverse, but you can still make a hot cup of tea. Or else you take a juicer, you put fruit from the top, you rotate the juicer clockwise, you get fruit and pulp at the bottom. Now you cannot say, I rotate the juicer in the other way, I invert the, the juicer, and I put juice and pulp, and I'll get fruit in another way. That's impossible. That's not possible in the real world that we live. There are reactions that work in when the delta G is very small. You by concentration changing, you can, by flux manipulation, you can have. But these kind of ideas of mechanical work, rotary work that spans multiple spaces, all that, this is just anthropomorphic extensions. They have very little foundations in reality. And most importantly, these are things that even a school-going kid can understand. First and foremost, an erythrocyte, it doesn't have nucleus. It doesn't have mitochondria. It sustains for 3 to 4 months happily. How can it? The glycolytic pathways cannot have millimolar levels of ATP. It is impossible. And also the control of the cell by genetic regulation, upregulation, downregulation, that's not possible because there is no DNA. And cyanobacteria, as I said, is too small. One of the biggest problem is a proton is 0.00002 angstroms. And an hydrogen atom is 2 angstroms. Imagine trying to control, specifically bind 0.00002 angstrom by a 2 angstrom minimally small atom. It's impossible. These ideas have very little foundation in reality. So aprotic environment, photosynthesis occurring side by side, proton being too small, and most importantly, the classical theory says that 3 sodium is pumped out and 2 potassium is taken in by this Jens Koos, who got a Nobel for that, sodium potassium pump. But then plants don't have such pumps. It is known. And then plants yet have 10 times more potassium than sodium. But the problem is potassium is a larger ion. Sodium is the smaller ion. So if you have a smaller ion inside, we can accept it through some kind of sieving. But we have the larger ion of potassium, greater amount. So the acclaimed mechanisms for cellular powering, coherence, homeostasis, electromechanical activities, sensing response are inadequate. That's the reason that I'm saying. Now, what we come to the Murburn postulates. Murburn postulates fundamentally believes that cells are crowded and stochastic environments. Diffusible reactive species naturally arise, and it is unavoidable that you can, stop it somehow. And system level control must be distributed, not localized. Molecules cannot be intelligent per se, but things can come together and they can be intelligent. And very importantly, some point I have missed out in this is cellular medium is not a dilute solution. It's a colloid. Anybody can verify this, but all theoretical treatments in classical textbooks consider dilute solution treatment for biological systems, cytoplasm. So before I go into Murburn concept, I want to just say, I want to pay tribute to Lowell Hager, with whom I worked in late 1990s, from where the journey started. Lowell had found this enzyme, chloroperoxidase, and his idea was the standing principle or mechanism of that heme protein. I went to his lab, availed his fellowship, worked on his NIH project, and opposed his idea. And he was so magnanimous that he accepted it after 6 years. It took him some time, but he accepted it. There you see, my tribute to you. You will live on in my memories forever, Lowell. And this is Stanislav Yanev. I was working on drug metabolism. I went back to US. I came to India to do social work. Then I decided that I don't have the ability to get my own following and people will not listen to me. So I wanted to make a big name for myself. I went back to US and I started working on another heme enzyme, the drug metabolizing liver microsomal cytochrome P50. There, I was in the land of opportunities. Thrice I was fired for holding on to my opinion. That cytochrome P450 is the proteins that metabolize the vast majority of drugs that you eat in the liver. All right. The liver is reddish brown because of these enzymes. Blood is red because of hemoglobin. Liver is red because of reddish brown because of these enzymes. So these, when I said my project, NIH project, was to minimize ROS production, reactive oxygen species production. But I reported that if I take out reactive oxygen species, the reaction doesn't occur. So what happened? I was fired. They said, you cannot continue. So this is the second system. Then I came to respiration, photosynthesis, multiple, then diverse metabolism, vision, mechanophysiology, immunophysiology, evolutionary biology, all that. And this person, Stanislav Yanev, he was also working in drug metabolism. All his work, he had worked through the classical mechanism. But when he saw my mechanism, he said, yes, this makes sense. And he supported me until 2024 when he died. To the two of you, my salute and my thanks. Before I move on, my gratitude and my respect to you. Without that, our society doesn't work. Okay? So first and foremost, I will talk to you about how I came to this idea of diffusible reactive species can give selective and specific reaction. First is the system that I was working on, chloroperoxidase. That is the fungal protein. Now, what has happened is usually if you take two molecules like thiourea and monochlorodiamidine, please be with me. Don't go too much with the molecular structures. It's not important. What is important for you to understand is two molecules were given in a reaction. Okay. And when two molecule is given in a reaction, and if I add a chlorinating agent like the toilet bleach, okay, both get converted equally, 1:1 ratio, quickly. Now, but when you give these two to the enzyme reaction mixture, this was consumed first. The sulfur lone pair was consumed first rather than the beta diketone. So the idea was the enzyme active site somehow selects this molecule over the other. And then when that is not there, this comes. Now, what I showed is you add the bleach in a small amount slowly, you get this converted. That is, sustained release of a radical or a reactive species can achieve selectivity and specificity compared to bulk. It's like I come through the door, and if I see Professor Raveendran sitting, I know I like him. I can, but if it is too crowded, my collisions with others will prevent me from meeting him. Otherwise, I'll spend more time with him because I know him more than anybody else. That's the kind of thing. There are even the diffusible reactive species at very small amounts, they have their own philia, they have own selectivity. That part is what I established. And also here, in the oxygen insertion reaction, you get substrate dependence, whereas the chlorination reaction is practically a zeroth order reaction. So here I said, this is because the radical comes out and reacts. This oxygen insertion at the heme center is through a diffusion diffusion controlled active site reaction. So I could show selectivity and all that changing by pH and chloride ion concentration. So how does this affect marine life and all that? See, chloride ion is high amount in life science, in marine life. So this will impact those ions containing medium more, because this kind of things afford greater amount of reaction impact with the ions. So what is Murburn concept? Coming to what is Murburn concept? Now you have a protein. This is the classical mechanism. This is an enzyme. You have an active site. You see the complementarity of the topology. The active site has a very beautiful complementarity with the substrate. So the substrate gets turned over like that. That is, another molecule that looks like this perhaps will not get reacted here, because that's how the active site is. Similarly, electron transfer also, there should be similar topological complementation. Whereas the Murburn model says you don't need such things. A reactive species can be generated which can react with a weakly bound non-topological complementary stuff also. So what does Murburn says? Murburn says that Murzymes are the agents that catalyze Murburn reactions. Extension of catalytic classical enzymes, they invoke diffusible reactive species or diffusible reactive oxygen species, or like what Muralidharan sir said, muod burning. It has one electron reactions. Usually one electron reactions are not considered to be very deterministically controllable. So our Murli burning, like what he said, and then molecule unbound ion radical. Our life is made of molecules, unbound ions, and radicals and radiations also impact. The components, they interact stochastically or not deterministically also. They can give also some reactions or mild unrestricted redox catalysis. This is based on effective charge separation. These are the terms. ECS to give cells that work as simple chemical engines rather than proton pumps, proton motive force, ion channels, specific, all that. These are the key ingredients here. Now, let us go secondary. This is a diagram made by Danny. He's sitting here. You see, here, inhibitor or substrate, they bind at different or the same place, binding-based outcome. So you have highly deterministic, specific redox partnering, integral stoichiometry. What is integral stoichiometry? A plus B gives 2C. But here, you see the Murburn reaction. This has gone. Power poi. So you see in that the reactive species, there is a zone around the enzyme where it can interact with various ions, various molecules, and then that can give altered effects of inhibition, activation, all that. So here you get broad substrate ranges, non-specific redox partnering, and non-integral stoichiometry. Just by one simple idea that non-active site reaction can give this kind of output. Very simple suggestion. So here what happens is, in this theory, you see how the middle one, you see, you don't need a complementarity because the diffusible species in the center, which is shown as stars, they can be produced and they can react with the final target to give the products. So this is what I say is the role of oxygen. Oxygen makes diffusible reactive oxygen species, and that interacts with one electron active species and all that. And there is like a whirlpool from which products come out. So here you have effective charge separation. Oxygen comes in, makes a charge separation, and it can give diffusible reactive species, which are not released at such like that. They are released in a sustained release dynamics manner. And then you have these two all together setting up a chemico-electromagnetic matrix, which gives the cellular intelligence, and PChems, which is also cellular intelligence, powering coherence, homeostasis, electromagnetic and mechanical activities, and sensing. So this is the way simple chemical engine works. So how do you control, like what Sir asked? This is basically thermodynamic control, pull, delta G of formation, dynamic partitioning, flux control, sustained release, concentration-based stabilization. If you have high amount of radical, they will coalesce. If you have smaller amount of radicals, they will be stable. But then there is proton availability, spin locks, all that. These are the ways in which you can control them. So at the outset, before going into any kind of application, you can see from a very conceptual way, oh, it's working. Suddenly I got it. So the first one says different topology molecule. This active site may be like this, but because a diffusible species is involved, different structures molecule can react. Then how different kinds of molecules with different moieties, you can get different kinds of reaction. By the same enzyme you can get. So earlier these were not explicable. And then the kinetics that you observed, That is, you have Michaelis-Menten kinetics, you have Km is equal to Kd plus K2 by K1. If you're an ardent student, you will know. That is, Km is a reaction constant, but Kd is a thermodynamic binding constant. Then how is it that sometimes Km becomes much orders lower? That's because Km is reflected by the diffusible radical species. So it can explain how you get various types of kinetics. This is very important. It has got medical applications. That is, in certain reactions, you might not get, in certain dose administration, 10 milligram drug, you might not get an effect, but 1 milligram you can. Why? Because at 10 milligram, you will have larger amount of radicals, whereas at 1 milligram, you will have very less. When that very less amount present, it is stable. Too much radicals, they all fight with each other, react with each other. They will destroy each other. So radicals, when present in a small amount, it serves just like in society, just like in chemical reaction media. All right. So lower amount, say, I'm not advocating homeopathy here, but then some drugs can be more effective at a dilute concentration. Then variable stoichiometry, there is a lot of difference fights. We get 2:1 ratio. Somebody says, I get 1.5:3 ratio. All that, 1.5:3.2. Non-integral ratios. All that can be explained by this new idea. Most importantly, how cellular electricity, transmembrane potential, that is, the classical theory said that 3 sodium pumped out, so positive ions were pumped out. Rather, this theory says oxygen comes in, takes electrons, becoming negative inside. So phospholipids contain this negative charge inside. So inside will be negative. You don't need to pump anything out to make the electrical activity. All right. So here is the way in which the pumping is replaced. NADH gives reactive oxygen species inside, and then that makes ATP. All right. So here you have in the proteins, colloids, they can bind to ions and co-solvation. We will show that potassium is more soluble in the cellular medium because of carboxylates, proteins, phosphates. Thermodynamically, potassium is greater stable, and the presence of diffusible reactive species inside the reducing environment thermodynamically favors potassium. It's not a pumping. People did not understand the thermodynamic foundation. That is why. So that's why the bigger ion of potassium is inside. So this theory predicts that DRS levels should correlate with function. Lipid phases should modulate redox functioning, buffering, and non-integral stoichiometry should be usually noted, which is the case actually. So we don't need pumps, and this says accepts colloidal nature, and it also, these are radical reactions which can be quicker. Genetic control mediated, all binding based effects are relatively slower. You cannot explain the kinetics. So this is beautiful in that way. So one example I will come to here is respiration. The most fundamental process of all living beings, all living cells, particularly aerobic cells. Aerobic is the ones that use oxygen. The classical theory, what Sir was talking about, is this. That is, there is the membrane, mitochondrial membrane. There are these large proteins, complex 1, complex 3, complex 4, that is complex 5. Now, they are supposed to be serving as part of electron transport chain, and there are diffusible species like ubiquinone or coenzyme Q or cytochrome C here. Okay. And that is supposed to relay electrons between these pit stops. It's like a manufacturing line. 1 electron goes, 2 electrons go from here like that. So that, and while that is happening, protons are being pumped. So protons can't be pumped because protons are too small to be held like that. There is no real gradient observed in physiology. And these are the reactions, and these are the kinds of ratios. So this is the structure of mitochondria. It's something like a hydroelectric power plant where you imagine there's a water at a higher level. It comes in, it sets a turbine going, and then you have somehow that is pumped directly. So protons are like water here. They are pumped continuously and ATP is made. This is the classical theory. All right. So what is the Murburn theory? Murburn says oxygen comes in, all those electron transport chain molecules, the proteins, they make these species radicals. Radicals attack phosphate and ADP. ADP is bound at those proteins that are on the membrane. And so the probability of a radical meeting the ADP and phosphate is high. If the protons are low, the radicals won't become water. They can react with ADP and phosphates. That is the beautiful reason why bacteria are 1 micron. Because only then protons will not be available. Otherwise, what happens is the radical will quickly take proton and become water. If proton is not available, it takes millisecond times to come in. Before that, it can react and make ATP. So these reactions are simple bimolecular reactions. Simple bimolecular. It doesn't require something 4 or 5 protein, each protein being made of dozens of genes to come pump all control across multiple phases. These are simple bimolecular reactions. Okay, this is the Murburn model. So this is the overall layout. That is, complexes 1 to 4 use different molecules. Now, sir asked why are they present? Now, complex 1 can use NADH, succinate dehydrogenase can use succinate, complex 3 can use so many things in the vicinity, and complex 4 can use peroxide. It depletes the peroxide completely to water. So all of them generate DRS and they make ADP and they react with ADP and phosphate to make water. And then the ATP formed is taken out by porters and water formed goes out through porins. And when this is happening, protons from outside come in. So these are all directional. There is no cyclicity in this. Everything is directional. Also, in the earlier one, you need to arrange the complex 1, 2, 3, 4 in a particular way, all that in particular ratios, which are defeated by the super complex structures that are available now. So here, you don't need any such arrangements. This new theory explains that multiple modality that you see mitochondria in. And most importantly, we showed in lab, you take ADP and phosphate, add superoxide, you get ATP. And this reaction is inhibited by cyanide experimentally. Very simple reaction we showed. And not only we, Kathleen Mailer had shown that in a mitochondrial suspension, if you take xanthine oxidase and xanthine, you get increased ATP. Xanthine and xanthine oxidase give superoxide. It's known. So also there were some other lady scientists who had shown like Mildred Kohn. In 1950s, she had shown multiple O atoms incorporated into ATP. That can only occur through radical reactions. So what are the evidence we have? More NADH and more oxygen increases diffusible reactive oxygen species. Increase in ROS. This is Nichols data from Aging Cell. He is the guy who has written bioenergetic textbooks. You see, when ROS is present, only when ROS is present, you see ATP formation. Only when ROS is formed, you get that electric transmembrane potential going to 200 millivolt. So it is the ROS that is doing. ROS is not a byproduct. They always thought ROS is a byproduct. ROS is not a byproduct. ROS is the fundamental agent. So ATP production is not seen without ROS. And complex 1 to 4 generate, all these complex 1 to 4, they generate reactive oxygen species. And all these pendulous regions are ADP binding sites. There is no role in the classical thing, at least for complex 5, there is. But in the other ones, there is no role, definite role. Okay. So I will come to two specific proteins. Some of you who are biologists can see this. Please bear with me. Here is some chemistry. Don't get lost here. Here, this is complex 1, perhaps the most interesting protein in the electron transport chain. This is the prokaryote. This is the eukaryotic in our system. This is in the bacterial system. They have the same structure. They have multiple iron sulfur centers and flavin at the top. And this part supposed to be the electron transport chain part. So they are disconnected, physically disconnected. And not only that, the electron transport is supposed to occur in microsecond time scales, whereas proton pumping is supposed to occur in millisecond time scale. There is no spatial, there is no temporal connection. And also, the Murburn theory beautifully explains why NADH can give that it gives only one proton, but gives 2 electrons. So we want a proton deficient system wherein the radicals can be stabilized. All these are deliberate ways. Why should you have all these ions? Why should you have all these redox centers here? If the classical electron transport chain was true, you should have redox centers in the membrane. So when the electron goes through here, protons could get pumped out. Most importantly, it is bioenergetics. The classical reactions, if you see, it's endergonic. See Murburn reactions. They are beautifully exergonic. They have high drives. If you see all medical biochemistry or standard biochemistry textbooks, the delta G values will be very small. These are very exergonic reactions. And all these prokaryotic to eukaryotic, we showed, Abhinav showed that they have high conservation of ADP binding sites. So these are the actual fundamental ADP binding and ROS reacting agents that make ATP. So this is beautifully shown. And then complex 3. In the classical theory, there is no role for this subtension. We showed ADP binding sites here. And all these, Peter Mitchell said CoQ cycle, Q cycle. All these hemes and ion cells, they are all solvent accessible. If there is any wiring, you would not imagine that. There is no way this can conduct any specific wiring. This is random, evolutionarily optimized way of using reactive oxygen species and coenzyme Q, that is ubiquinone here. What happens? This is in Murburn system. This is a radical scavenger in the membrane. It doesn't need to take from one place, from complex 1 to complex 3 like that. This is immobile practically. It's too big. And so what it does is there are different kinds of lengths of these things. They are packed in the membrane. And why? So the larger one is the more predominant one. If you had this serving as a transfer agent, you would imagine the smaller one to be preponderant. But no, otherwise. So this is the new theory. You can see, like earlier, all the Murburn reactions are exergonic. Thermodynamically facile, kinetically facile, agrees with the structures, all that, and agrees with the inherent nature of mitochondria. This is a work that Danny and I, we had predicted in 2018 in biomedical reviews that thermogenin, the protein that is supposed to generate heat in the body, in mitochondria, we studied the protein, whatever structure was known. We said it will have high lysine and arginine in the interfacial area that will enable the reactive oxygen negatively charged. If you have lysine and arginine, they are positively charged. So they can move things better. They can facilitate proton dynamics better. So if you have proton dynamics facilitated or movement of anionic species, they're also called anionic transport agents. Now, if they can move it, then heat will be high. So we predicted that the structure will be like that. True to our prediction, in 2024, it was revealed that it did have. So we sent our paper, letter to the editor, to Science. They published it online. They can offer rebuttal. After 2 months, they took it off without even informing us. The editors didn't offer, or the authors didn't offer any rebuttal. That is a Nobel Prize winning people's group. So that's why we should have some mechanisms to advocate our own science. So the classical theory required fatty acids to say how the classical theory said this proton gradient is dissipated with using fatty acids. There were multiple theories. We don't need all such theories. We can explain heat formation in body beautifully based on radical reactions. Their kinetics are fast. So when radical ROS react among themselves, they make water and produce heat, and that leads to thermogenesis. Beautiful structure and function, beautifully explained. So now this is a macroscopic view. I still have about 10-15 minutes. We'll finish in 1 hour. So the idea is here, that here it's like a grill. You know, some heat source happens and some material is placed and some reaction that gives steam, smoke, you have cooked food. So it's a thermal, a very simple chemical process where you can draw analogy. That is steam, like you have a combustion reaction, carbon dioxide, water, all that you can find analogy. Unlike in the classical theory where you had redox process disconnected from the phosphorylation process, this is a directly coupled bimolecular reaction that's overall highly facile. So it explains transmembrane potential, thermogenesis, Turgor movement of water explains homeostasis. So this is a fortuitous assembly, independent and parallel functions, agrees with the architecture. Overall, this is very simple. So whole perception changes, fatty acid giving rise to toxic signaling and all that in heat and all that here. From here, the whole chain of understanding changes. So this is a holistic idea. This is something that I was talking to our sir, Vasudevan sir. Only 2 months back, we nailed this. That is, Krebs cycle is another reaction that is happening in the cell. After glycolysis, acetyl coenzyme A, it is making the NADH. Now here, I analyzed the thermodynamics of this reaction, whole cycle. I found it to be highly endergonic. It is 10 to the power, the K equilibrium constant comes to be 10 to the power minus 127. 10 to the power minus 127. No way Krebs cycle reactions can occur if the Murburn reactions don't occur. So the carbon dioxide releasing function can occur only with oxygen. That is beautifully explained by the Murburn view here. Here, it requires NADH to be made. Krebs cycle cannot work without this happening side by side. So rather than a parallel reaction system, this is like a cauldron where both occur simultaneously. So the thermodynamics, kinetics, feasibility, everything in mitochondria, this is conclusive argument. I gave it to ChatGPT, I gave it to Cloud. They finally said, this is too much for anybody to escape. So this only 2 months, we have not yet published it. So this meeting has several unpublished release of ideas. So that's what a seminar is supposed to be. Meeting is supposed to. So this is something somebody should have come online. He is not online. Paul Gagnew's idea. I went to Bulgaria this March. I was invited there to present. And this is, he was studying the dimensions of mitochondria. And somehow, here it is the mitochondria thickness, mitochondria length. He presented one data and he said, there is an area I don't find mitochondria. Very simple. Here, pi r squared h is the volume of mitochondria. If your thickness high and length also high, the volume is high. When the volume is high, protons are more. If protons are more, it cannot function. Very simple. Very, very simple. So he bought it. But he didn't come today online. He said he will. Then red blood cell, another application. I told you how can red blood cell sustain. Red blood cell have hemoglobin, perhaps the most abundant globular protein. It's a tetramer. It has 2 alpha and 2 beta monomers. The classical theory has nothing to do with beta monomer. It is just like alpha monomer. But you see, 2 beta monomers, they have got, this is the way oxygen goes in, and the other side has a small hole. In the tetrameric tense form, Abhinav showed that it binds to ADP, and when iron 3 releasing superoxide or iron 2 releasing oxygen is the same. Both can be possible. So when it releases superoxide, it can make ATP. This alone explains how you can get 3.5 millimolar of ATP in red blood cell. So you can get how maintaining high amount of phosphate always inside gives more potassium inside, homeostasis and all that, energetics. So overviews, we are coming towards the end of my talk now. I'll finish it in 1 hour as expected. Overviews. So now we understand why the rod cell have high packing of lipids. Mitochondria is like this. Same thylakoids, cyanobacteria, liver microsomal, all these places where reactive oxygen species are high. Why do we have high lipid packing? Why? Because water should be minimal. If you have water, you will have protons. So that is why. Then we showed that in all places, Murburn reactions occur. Then we showed that post-translational modifications are not just post-translation, not just at genetic level, at a metabolic level also, you have multiple kinds of impact by Murburn chemistry, radical chemistry. That's what is shown here, post-translation. So you come to an overall macroscopic and molecular and conceptual understanding wherein molecular level, diffusible reactive species interact with iron and all that, and they can make chemical processes. At a molecular level, these chemical processes can give charge, heat, pressure, because water is formed, gives pressure. So all these things like a simple combustion engine, you don't need pumping, you don't need molecular level intelligence. So it works at a systemic level like that. And so at a conceptual level, short-term idea is Murburn concept, stochastic, and then deterministic information, long-term, you give cell. So here, diffusible reactive species, effective charge separation is the fundamental foundation. So now our understanding of overall conduction of earlier, you see, you would imagine it's like a classical orchestra where everyone is given a piece to be played in a given time. But then a jazz band works in a different way. People have an idea and they just play along based upon each other. This is the new idea that you get. It's not that deterministic. So now here you understand why oxygen became so important. Oxygen can directly enter into equilibrium compared to those sulfate, nitrate reducing bacteria. Here oxygen, you can exploit the full -400 to +800 millivolt full range, and you can get that. The ions are electrolytes and lipids are proton limiting environments. Proteins, they have active site specificity. Okay, carbohydrates. Why carbohydrates form the basics of nutrition? Because OH groups, water, hydroxyl, that is the way it has to go. Nucleic acids have phosphate and OH groups there. Why? To prevent the ROS, to attack the bases. So oxygen needed for generating ROS. So from this, you can have stochastic spontaneous interactions. You can have emergence of biological intelligence through PCAMs. So here is a new concept where spatial probability, Murzymes, these were the primary enzymes, and classical enzymes are when the transition state and spatial reaction sites are minimized, you have the classical enzymes. The Murzymes are the largest set of enzymes. Murburn does not negate classical mechanisms. It complements and extends them. This is a misunderstanding that people have. All right. So here is the basic idea that people can very easily relate to. That is, if you find a knife in kitchen, it is also associated with cutting board and knife racks. When you find a knife in kitchen, you don't say, oh, knife is not to be used. Similarly, if you have antioxidant enzymes like catalase, peroxidase, superoxidase, it doesn't mean that reactive oxygen species are bad. It means that we need it, but it's like knife. It has multiple problems. So it can give order, it can give chaos, it can give both. So these are the elixirs of life. They explain, science is about, physics is about energetics, kinetics, mechanism, structure, distribution, evolution, probability, Occam's razor. All this Murburn explains better. Only thing it doesn't explain is the stigma or aesthetics. What if? Sir said, how do you get the control? There are control mechanisms, but then it is in a sustained way. We have to understand how all this happened now. So it is, we studied in school that the difference is like this between combustion and respiration. I say respiration is also very much like this. And in the Murburn school of thought, ATP is important, but it is not the complete. So very important. Why do you die? Because fundamentally, you are like a cloth that is dipped in oil and set fire to. Now, the oil burns, the cloth will also burn. But the oil burns first. So you are born, you will use oxygen like this, you will die. It is destined that you will die. It is not a toxic side product. Murburn dictates that you die because these are the imperative agents in life process. So oxygen has this Dr. Jekyll and Mr. Hyde, or the Batman comics two-face, you know. It is not, the classical theory says oxygen goes and binds to cytochrome oxidase to receive 4 electrons and 4 protons to make water. It's not the way. The workers of life doing chemical, thermal, electrical, mechanical, all these works are the diffusible reactive agents. They connect things, they react, they destructure water matrices. We are composed of 70%, 80% of water. All this structure, destructuring, when we move this, all this is happening because of water activity and not just protein. We have to see beyond all that. So that's the role of oxygen. That's what we have to start seeing now. All right. So impact, you see, this video has to be played. Can somebody play this video? First, it's getting recognition. Is there audio?
Again,
That's George Chaldakov. I was invited there.
our society elected you honorary member of Bulgarian Society for Cell Biology.
Thank you.
This is a little title you. This is for you.
Oh, that's my culture. That's George Chaldakov. I published first this Murburn model of bioenergetics in his paper, 2017. Next year, Scopus Index gone, PubMed gone. Next year, the very next year. But every year after that, he has put Murburn on the cover page. He was supposed to come online. He is not able to. He gave the message, perhaps we'll play. So what's in it for clinicians? I say that Murburn is the, perhaps in due time, it will save more lives than any other quantum of information. It will prolong life. Now we are 60 to 70. We will live to 100 in 20, 30 years. And it will enrich our life more. So I don't want to go on now because we have to have discussions also. It's already 1 hour. So here you see what is the impact. After the publication, proton motive force and chemiosmosis is going down. Mitochondrial work is going up. Recently, just this month, we saw pharmacology reviews, 20 impact factor, Chinese people citing 5 papers of Murburn in mitochondrial metabolism. So this paper, Chinese group, again, 18 papers on Murburn. All right. So another one on hepatocellular carcinoma, 5 papers on Murburn. All right. So this person has given a message, Marko Fiore. We'll play it later. So I would end by saying, we, in our culture, we called digestion assimilation process Dahanam, Dehagni, Kayagni. We call it. Why? We have our body filled with. I think it is not really great to dismiss all ancient ideas and terms. Just like that, Shante Agnom Riyate. There's a fire burning inside. They must have had some intuitive idea. It's like burning. That's why they used it. I also say that. But I got to know about it only now. So this is my favorite comic by Bill Watterson. It's a magical old world, buddy. This is a 6-year-old boy talking to his tough tiger. He says, let's go exploring. White canvas. Always, if you are young, budding scientists out there, don't take anything anybody tells you unless you try to understand. All right? When you try to understand, when you ask questions, then is where you start becoming scientists. These are questions. These are all questions I have prepared. Okay. Thank you so much. I thank, before I, I think Professor Babu, Professor Vitas, and Rajendran next session, no? He is next. He can call him here also anytime. No, Professor Babu, he has been with me for a long time.
Okay, then
Professor Babu, can you please say, or Professor Vitas?
Yeah, okay.
Professor Babu, please go ahead.
Yeah, can you hear me?
Yeah, we can hear you.
Okay. All right. Thank you for inviting me for this special presentation. It's kind of late here, because Earth is a round object. Time here is 2:50 a.m. But, I kept awake so that I could listen to Murali. I have known Professor Murali since the early 2000 when we both were working as postdoctoral researchers at the University of Illinois. And I have seen firsthand the inertia shown by some scientists to have an open mind to ask logical questions about these classical models and how they compare with the Murburn model. However, the past couple of decades have witnessed a steady progress in the research and the reach of this model, as Professor Murali has correctly described. It's not just a simple model or a new theory that explains some few processes like cellular respiration. It indeed is a paradigm shift with far-reaching implications for our understanding of life and its evolution. So I would like to make two important points here. One, the scope of Murburn model is very broad. Professor Murali has already shown that it can be applied to a number of phenomena. He has explained the mechanism of vision, how very little amount of cyanide can kill a person, and so many other examples. So a lot more research can be done in this field, and many physiological processes can be looked at from the perspective of Murburn model, and this will certainly lead to new advances in both basic science as well as applications in the field of medicine, etc. Now, I'm a physicist, so I would like to make one important point. We all know that living organisms, starting from, say, the single cell forms of life to the apex predators, they represent thermodynamic systems. Thermodynamics is one of the oldest and most fundamental branches of physics. The laws of thermodynamics are fundamental laws of nature. Living organisms represent complex thermodynamic systems. The degree of complexity increases as we go from, say, single cell organisms to more evolved animals. Now, as far as thermodynamics is concerned, no matter how complex a thermodynamic system be, it must still obey the fundamental laws of thermodynamics. The Murburn model is a theoretical framework built in accordance with the basic laws of thermodynamics. So that is one of the most important reasons why this model is going to prevail and emerge as a basic principle of life and its evolution. Thank you.
Yes, thank you so much, Babu. And you were witness also to the fact that Lowell said that these people will not accept it. You remember we were in Urban Summit.
I remember that very well.
Yes, thank you so much, Babu. It's late. I know, please. Thank you so much. Thank you. Yes, thank you. Professor Vitas, please. Dr. Vitas, Marko Vitas, can you?
Yes, I can hear you. Yes,
Yeah.
Good morning.
Yes, good morning. Vitas and we got to know each other after my research, and we interacted over. He's an evolution specialist. So we know each other only through work. We have not collaborated. He's an independent researcher. Please, sir, please.
Thank you very much for this very interesting lecture, beautiful lecture, where the whole complexity of Murburn concept was detailed, and I would like to point out that iron sulfur are very ancient enzymes, and this Murburn concept will contribute to understanding biocatalysis and evolution of biocatalyzers, and also consequently would impact our understanding of origins of life. So may I ask you a question?
Please do, Professor Vitas. Dr. Vitas.
I would like to ask you, what is the most provocative part of your concept in your Murburn concept? What is the most provocative which you found difficult for a broader scientific community to accept it?
The most provocative is the one that Professor Vasudevan said. The third part, actually the second and third part are part of the same question he asked. He said, how do you achieve control? Because you have a radical, oxygen radical. Currently it is seen as something that creates chaos, creates diseases, is resulting in aging, all that. I don't say that it doesn't, but then it has a good side. So to get people to go beyond the aesthetics, beyond the stigmatic perception that prevails, that these reactive species are bad. I don't say they're not, I don't say they're not, I mean, I'm not getting the logic right. I say they could be bad, but the case is that everything in life has got a context. It has to be in the right amount, at the right place, at the right time. Three things have to come together. So what I say is reactive species at the right place, at the right time, in the right amount is good. It can give practically zeroth order reaction. So something changes, this will keep it stable. You want something to change with change, but you want something to be stable also. So radical species at micromolar, submicromolar level is good, is something, the most radical idea that is difficult to sell. And of course, every other aspect of Murburn is basically, if people have something, it is making it opposite of whatever people believed in. So it's very difficult to get people to accept that. They have projects ongoing, they have PhDs ongoing, they have published textbooks, but then there are great people who introduce these new ideas. If the young people get to see the new ideas, they will naturally see that this is more conducive to evolution, more conducive to probability, more amenable to structure, function, distribution, thermodynamics, kinetics. And that's what we try to understand life with. Thank you. Anything else, sir?
Well, thank you very much.
Thank you so much for staying online for so long. Sorry that it got a little delayed. Sorry.
Okay.
Thank you. Yeah. Professor Babu was from Western Illinois University. He's a professor of physics. He's in US. I think those people who are online, please excuse because it's already become late. Sir, please.
And Rajendran will speak in
next session. Who else is online? Mahendra Kavadiya can use another session. Tofia Hussain is my collaborator. So those two were guests. They have not collaborated with me. So who else? I think Professor Sukumar also can do. Mahendra, hi. We can talk sometime later because I think it's already 1:30 and people will have to go for lunch. We will hear all. Mohan sir, please bear with us. So if anybody wants to say something, they can say, but otherwise it's okay, I guess.
Sir.
Yes.
Murali,
yes.
sir.
Yes, please go ahead.
Hi. So, sorry, I know that it is late, so it was a very nice presentation. Second time I am listening and the lecture, last time it was in CPCRI. I have one quick question. Please. Did you do any, first, initially I didn't listen, so that's why. Did you do any experiment using the isolated mitochondria? And the one you might have seen, there is a classical experiment where isolated mitochondria, just they put it in two different buffer system, by changing the pH and where valinomycin they put, and they simulated an experiment and they could generate ATP without substrate actually. So did you do any similar kind of experiment so that it will add more, support? And being a teacher who teaches this particular topic for my biochemistry student, being a biochemist, so which direction? Of course, it's a very novel thought, no doubt in that. As Vasudevan sir mentioned, I think I didn't hear at that time, the control of this ATP synthesis is becoming a big question there, because mitochondria is very sensitive for the free radical. So cytochrome C will be going out and cell will die if there is a imbalance in the free radical content of the mitochondria. So that's why I have this particular question. Of course, we have a lot of questions we will discuss another time. I know that it's very late.
No, sure.
So
this is a very important question, because Jagendorf's experiment with chloroplast, that is what triggered all this. When Peter Mitchell proposed that proton difference gradient is what gives rise to ATP, they did not really understand the thermodynamics behind it. And they did an experiment, and it was false interpretation. I'll tell you why. Now, H plus ADP plus PI plus H plus gives ATP plus H2. That is the actual equation. So in that experiment, what they did is that they gave surplus H plus in this surrounding. So that will thermodynamically, because H plus contributes in this equation 452 kilojoules per mole on the left side. When it gets on the left side, you have delta G formation calculated by the right side minus left side. So 452 joules on the left side you get highly facile ATP synthesis. But that has no relevance whatsoever to physiology, because in physiological system, there is no pH gradient. Inside is the same as outside. So now if I take a lion, put it in a circle, circus cage, and make it jump through some fire hoops, and I say, in reality, it is in the jungle also, it lives like that, it has no relevance. So, Britton Chance very clearly showed in 2 Nature papers, when Peter Mitchell proposed that this proton gradient is what is giving, he showed very clearly there is no pH gradient. I don't have the expertise or the experimental setup to do, but I can easily rationalize it. My cover page credited paper in Progress in Biophysics and Molecular Biology clearly explains these experimental results. Why the bioenergetics community went on the wrong side? Because the H plus contribution was not really taken. That's the reason. So that doesn't have any physiological, and they forgot one thing. They did that experiment with rhodopsin and complex 5. And, after that, first just with mere mitochondria, they did. And then they showed incorporating rhodopsin light-induced proton pump. But they did not realize when they lacked complex 5, rhodopsin alone gave ATP, 20%. That data they completely refused. So Murburn is a more complete explanation, which explains what complex 5 does. Complex 5 uses ATP and opens up the internal. See, when you have a stove, chula, when it gets stuffed after a particular time, you need more air to come in. That's what it does. It opens the, uses ATP, opens it, lets the outer protons come in. When protons come in through solvent, then the bound ADP on that use phosphate and can also make ATP with that. So that explains how ATP is made.
I think you'll have to have a lot more discussion on these things anyway.
Yes. Okay, thank you.
Last question. Wonderful question. Shall we have one last question from here? Anybody?
Questions have prizes at the end of the day. Krishnamar sir, namaskar.
No, but there are no questions. There will be lunch.
Please ask questions. How many of you have studied this? Respiration? Yes. I taught you guys. So the boy raised a hand and the girl asked him to keep it down. Yes. Chairs, any questions from you, sir? Did I address your questions? Sir asked me 3 questions. what is the purpose of ETC? I showed you, perhaps that would have answered. The aesthetics control, I posted a slide, it had some content. That I hope. Yes, Ravi. Yeah, sir.
In the science, there is a general concept that when there are two opposing theories, and one theory is very, very complex, and the other theory is very straightforward, then usually we take this straightforward theory. The moment that he tells us, a few years back we met, and he explained that, then the simplicity of the theory is what attracted me first place. That it is so natural, a natural concept is there, and that is what attracted her, and that is why I took it to my textbook also as an alternative theory. Now, secondly, the photosynthesis, the main point I wanted to tell that the RBC, even from my undergraduate days onwards, I was asking myself and all my teachers and colleagues, etc., how the RBCs are there for the 120 days lifespan, whereas there is no enzymes there. Enzymes are there, but there are no DNA, there are no protein synthesis, no ATP synthesis. The whole the ATP synthesizing machinery is not there as per the current theory. Therefore, how it is working like that, and there is no real answer for that. Therefore, this is an answer, that a direct answer that he can give, even though we have to experimentally prove that, that is another matter. But at least a theoretical concept is coming here, and that is a great point that I am taking it as a point that is pro for this particular concept. And lastly, I wanted to tell that you have to change the Murburn concept to Murali's concept, because otherwise, Murali's concept, because otherwise after 10 years, some Westerners will take that it is Frederickson's concept.
Sir, yes, sir. Sir, I wanted to live beyond my name. So I have burnt my whole life, 25, 27 years now. He said, no, Murali burned. So, but then I have commented, sir, when Peter Mitchell can say chemiosmotic, which doesn't have chemistry or osmosis inside, and when we can all say it and teach it, I have 5 reasons to give Murburn. Molecule, unbound ion, radical radiation, mild, unrestricted. I thought a lot. My name, today I'm here, tomorrow not there. When I was in 12th class, sir, I wanted, when I would read all these named reactions, Canizzaro's reaction, Einstein equation, I wanted to have something with me. We have kids, we have work, then we have ideas.
I just want to have a, something. I must admit that I haven't understood much of it. But one thing I just wanted to comment on is some of these things which you say at the molecular level, I think we still have to go down to the molecular level. We are still talking about the macroscopic models instead of molecular levels. For example, I still remember one paper by T.G. Sparrow, a resonance Raman spectroscopy paper, where actually he discusses about there is a lot of coordination chemistry and so many things which are involved in it, where he was actually talking about the movement of this, the iron porphyrin system. And he was talking about the thing going from square planar to pyramidal symmetry.
Yes.
You can actually see that molecular level. And there is actually a lot of coordination chemistry that is involved when you talk about oxidation, deoxygenation, and all these things when you talk about, which I do not see in these kind of a macroscopic description. Probably that is what will have to get added up in the coming years.
Absolutely. Sir.
I follow him that I could not follow many of the things. Naturally, it is complex, but there are certain name questions to ask you. You were talking about the size of a proton. Okay, the size of a proton or negating something. That is in positive side or negative side. For example, size of a mitochondria. Okay, up to that level it is okay. But if you try to explain on the basis of the size of the proton and arriving at a number 6 there, I don't think scientifically, for example, the size is considered for a particle. In a proton level, you say that, okay, this is a particle and it has a size. It is difficult for me to immediately think that proton as a particle. Okay. So relating to the level of a proton, electron, their size to explain an observed phenomenon that it is getting too congested, how it is possible, etc. What is the rationale of this type of a correlation? This is a one question from my side.
Sir, that size
is the question. Is the question clear?
Wonderful, wonderful, sir. So the size is not the one used for calculation. The calculation is based on thermodynamics, basic. The dissociation,
etc. You did a multiplication.
Yes, sir. Yes, sir. That water dissociation equation, the pH measurement very clearly says H plus concentration is 10 to the power minus 7 to 10 to the power minus 8. So the molecule, the concentration is that much. So you multiply it by the volume, you get the number of actual protons that can be theoretically present in that volume. So that's what is the number. So the theoretical calculation is absolutely right. In the sense, the dimension is used for something else. The dimension of a proton is known. It is approximately 1 femtometer or something like that level. No, but then protons don't exist like that in reality. Protons exist as hydronium ions, Zundel, Eigen, different levels of hydration. Now, but when biochemists or biologists use protons, they speak like proton stands there H plus, the glutamate takes H plus like that, they show it like that. But if they exist as hydronium or their hydration is important, they should also try to talk about the hydration energy, the size of the hydronium ion or Zundel ion or Eigen ion, all that, which they don't do. They conveniently pass it on. My point is, the size of proton is so small, individual, it is impossible for ions, larger atoms to selectively bind it. They say selective binding and pumping. That's what I'm questioning, because
that's a kind of different description altogether, because I don't think there is any proton sitting as such. So when there is nothing of that sort, there is no scope for such discussions. But proton transfer in water particularly, there are beautiful ways that people have shown. You give a proton here and take the proton there. It's almost like a Havala racket that
Grotus mechanism. It is called Grotus mechanism. Yes.
Yeah, that's where in biology there are a lot of beautiful things like with respect to proton or electron transfer and how the chains, these proton chains actually assist in that. Anyway, I think it was a wonderful discussion and we will wind up now. For the time being, I think the lunch is waiting for all of us. And thank you, Dr. Murali, for a beautiful presentation. It was a beautiful presentation. I hope you have a beautiful concept that you have put forward and you will have the questions that comes out of that and answers that are coming out from your side or somebody else's side in the coming years. Eventually, in science, things get ironed out over a period of time. So thank you very much for a wonderful presentation.
Thank you, sir. Thank you. Thank you, sir. Vasudevan, sir. Thank you, Vincent, sir. Thank you.
Yeah. Just as a concluding remark, I would like to say, kids are playing with the building blocks. So if the building block is having a 10-pair length block, we can put another 10-pair block about it, just like in the lock and key arrangement. But there can be individual 10 one-pair blocks also, 5 2-pair blocks also, 3 plus 3 plus 3 plus 1. So there are different mechanisms. So in nature also, like we have seen only the 10-pair blocks, there are much more ways to resolve the things, what is happening. But when we study science, we generally try to look into the significant correlation with the cause and effect factors. So whatever 90% goes, we feel that it's well and good. So we establish such theories. But in all the cases, when this 90% factors holds good, we can standardize those things and look for the 10% thing. So same way, we have to still work on refining the ideas.
Definitely, definitely.
Standardize those things, bring this 10%,
Definitely.
100, then
Definitely.
look into more possible ways of
Definitely.
doing it.
I agree.
So all the building blocks can be properly inserted. I also admit, along with my co-chairs, I also partially understood what you were saying. There are different mechanisms of resolving some issues. My biochemistry starts and ends with Leninger. So not beyond that. So thanks for this wonderful lecture. I thank the panelists who joined with us, Professor Raveendran, Professor Damodaran Vasudevan, and Professor Venugopal for chairing this session, and Murli Manoj for Kerot Murli Manoj for opening up the ideas on Murburn.
Thank you so much.
Let's have more vibrant discussion. Lunch is arranged at the ground floor. Ground floor extreme end, there is CMFR canteen. You have to go to the ground floor, go to the northern extreme. There is a CMFRI canteen in the ground floor. Please join for the lunch. Thank you.
So from the organizer side, I thank all of you in one word. And those who want accommodation tonight and not yet obtained the key, please go to the registration counter and interact with Mr. Ramesh. I think some of you want already asked for accommodation, but not reported to the accommodation place. So you can collect the key from Mr. Ramesh. He is sitting in the registration counter. And we will start with 40 minutes lunch break. I request Professor KMM to do the duty. And we have with us online Dr. N.H. Mohan. He is the director of ICR National Animal Genetic Resources, National Institute of Animal Genetic Resources. So I welcome online, sir. I welcome you. And I request Professor KMM, please, to introduce the other speakers and panelists.
Hello. Afternoon. We move on to the second session, primarily biochemistry and the logic of Murburn. So we finished in the starting, we saw what was Murburn concept, how is it different from classical mechanism with respiration and thermogenesis and red blood cell as an example of application of Murburn concept. In this, very interestingly, very importantly, how Murburn concept allows us to explain in biochemistry, the most important way in which we probe the mechanism is by using toxic principles. So cyanide is a toxic principle which was understood in a particular way. My student is currently doing PhD with me. He actually did his master's in 2008, 2010. And now he is doing part-time PhD with me in ethics and the issues associated with Murburn. He will talk. His name is Vivian David Jacob. He is from Kottayam. He will talk now on cyanide. Vivian David Jacob, I said. Vivian David Jacob, please. The floor is yours. The chairs want to sit there, so I think it's okay. Sarah Kande. All the people are sitting.
Hi, everyone. Good afternoon. My name is Vivian David Jacob, and today I'll be presenting on why cyanide is so deadly. We'll be covering what the classical theory says about why it is so deadly, and we will also be talking about how the Murburn concept can better explain why it is so deadly. And I think being in Kerala, we might be very familiar with cyanide of late. If you followed the news for the past 1 or 2 years, there have been several cases where people have used cyanide to poison others. So why does it kill so quickly? So what does the classic textbook theory talk about cyanide? The classic theory is that our cells make ATP using an enzyme called cytochrome oxidase. And this has iron atoms inside it, heme centers. And what the theory says is that the cyanide sticks to it or binds to it like a lock and key model, classical binding mechanics. And then since the cyanide goes and binds there, oxygen can no longer bind and make ATP, and the cell eventually dies. Sounds like a very simple explanation, doesn't it? But when you go into the details of it, when you add up the numbers to see whether this actually explains it, it doesn't really add up. Let's look a little more closely. Okay, does it sound really right? So if you calculate the sites or the cytochrome oxidase sites specifically that are there in an adult human being, we can see that it's approximately around 10 raised to 22 sites that are there in an adult human being in cytochrome C oxidase when you calculate the number of cells, etc. And how much cyanide is needed to kill is approximately 15 mg. That's a little dose, which is about 10 raised to 20 cyanide ions. Or let's say 15 mg is approximately the weight of a housefly. It's a very small amount. Now, 10 raised to 22 sites versus 10 raised to 20 cyanide ions, when we look at it, it might look like a small difference, but it is actually not. Look more closely, this is how it looks. You can imagine one cyanide for every 100 sites to bind. So if you look at the red dot there, which can bind into any of those square-shaped crevices in these blue circles, that's it. Or for fun's sake, if you can imagine there is one fly in this room and it has around 100 of us to sit on. That is how, when we calculate the number of binding sites versus the number of cyanide ions that can kill. So the first thing is the classical theory. The numbers don't really add up in terms of the concentration of the dose, lethal dose. Now, another thing which we can look at is how tightly it sticks. So if you look at the KD value is just in enzyme kinetics, it speaks about how tightly one molecule binds to the other in a reaction. So if you look at cyanide and the values of carbon monoxide, you can see that it's much, much higher for carbon monoxide. It's 4 into 10 raised to minus 7 and 6 into 10 raised to minus 4. Thing about KD values, the smaller the KD value, the tighter the binding. So ideally, as per the values, carbon monoxide should be more stickier to COX than cyanide. If you look at the IC50 values, again, you can see that lesser amount of carbon monoxide is actually required for lethal dose than cyanide. And then again, if you want to look at the speed of it, how fast it actually attaches, you can see that carbon monoxide is almost 800 to 1,000 times more faster in terms of attaching to COX than cyanide. So why is cyanide more toxic still than carbon monoxide when it comes to it? If you look at the binding data, it actually suggests that carbon monoxide should be more deadlier, and followed by hydrogen sulfide and followed by cyanide. But in real world, what we see is that cyanide is more toxic, followed by hydrogen sulfide, followed by carbon monoxide. So cyanide has weakest, slowest attachment rate of the three, yet it is more toxic. So let's assume toxicity might depend on something else. Let's say speed. Let's look at the speed of it. I'm not going into the details of the calculation here, but if you look at the speed at which it binds to COX, you can see that it's about 1,000,000 times faster. Cyanide actually binds 1,000,000 times faster to ROS. So here I've shown ROS or reactive oxygen species. We'll get to why I'm showing reactive oxygen species in the next slide. But if you look at how cytochrome oxidase and cyanide binds versus cyanide binding to ROS, it's about 1,000,000 times faster. So we've dealt with concentration, we've dealt with the tightness of attachment, we've now looked at the speed of it. Let's also look at some theoretical calculation, dynamics. So this is basically the equation that you see there is basic binding kinetics. If you look at it, you can see that the KM402 at COX is very, very small, whereas the KD is much, much, much higher. This Km should actually be a bigger value than KD, according to classic kinetic theory. But in terms of cyanide and the classic explanation, if you look at it, it violates that law. So that's a red flag again. Now, let's look at what the Murburn idea says. So in the morning, you would have seen that what Murburn concept says is that ATP is not actually made by the oxygen going and binding as per se to cytochrome oxidase. ATP is made first by the enzyme which makes ROS or reactive oxygen species like superoxide, etc. And then these reactive oxygen species make ATP, or they catalyze the reaction to make ATP. And then what cyanide does, or this Murburn concept's explanation of what cyanide does, is that the cyanide actually goes and binds to ROS. And then it creates futile ROS cycles where instead of making ATP using ROS, it just makes water. So with ROS being, the cyanide causes ROS to be constantly used up. In the previous slide, when we looked at speed, we saw that it's about 1,000,000 times faster than oxygen binding to COX. So that's what the Murburn idea says. And finally, let's also look at thermodynamic feasibility how Murburn can explain this. So when you look at the reaction between oxygen forming superoxide, it's about 250 kilojoules, negative 250 kilojoules per mole versus negative 275 kilojoules per mole for the reaction of cyanide. It might look like a 10% difference, but it's not actually 10% difference, just that 10% difference. What it means that this cyanide reaction with superoxide is actually 24,000 times, because it's calculated on a logarithmic scale. It's almost 24,000 times more feasible of a reaction. So here we'll just, to summarize, the textbook explanation says that it binds to cytochrome oxidase, cyanide binds to cytochrome oxidase, preventing oxygen's accessibility. Now, 1:1 stoichiometric physical interaction. It's a classical site competitive inhibition. What does the Murburn concept says? Cyanide acts as a non-competitive catalyst, which explains for the very lethal dose at very small dosage. It reacts with diffusible ROS, which we saw that is thermodynamically feasible, which we saw that it's 1,000,000 times faster, etc., thereby disrupting ATP synthesis. So to sum it up, classical theory does not explain such a small dose. The classical theory does not explain the speed at which, within seconds to minutes, people die with cyanide. It does not explain that. And it does not explain why other molecules with stronger binding are not as toxic. It does not explain the deviation from classic kinetic equation. It also does not explain the thermodynamic favorability of the ROS reactions with cyanide, etc. Whereas Murburn concept is able to explain all of those. So thank you for your patient listening, and that's it from me.
Next, we have, please note down your questions if you have. Next, we have Dr. Prasad Burra, VLS Prasad Burra. He is a professor from KLEF University. Guntur, right, Prasad? Yes? Yes. Prasad got his PhD from IAC. He is into a lot of bioinformatic and computational biochemistry kind of work. And he is going to talk about a recent aspect that we unraveled. Most of it is not published. It is available as a bioarxiv preprint. So Professor Prasad Burra, please come. Thank you.
Okay. Okay, I am Dr. Burra Venkatalakshmi Siva Prasad. So I don't mind. I'm a structural biologist from IISc, Molecular Biophysics Unit. Before that, I was at University of Hyderabad. Wherever some idea or innovation is needed, I generally get carried away with those things. So currently I'm working on a new methodology in vaccine design and in methyl transferase drug designing aspects. And also I am also into healthcare products. So around 5 patents I filed looking for some investment and things like that. And that healthcare thing is how Ayurvedic marma chikitsa based things can be used to develop chappals, which automatically will take care of your health. So that's what is one more concept. So before getting into Murburn concept, if you have observed from morning onwards, we are talking about nanoscale and angstrom level mechanisms. And my PhD was also somehow related to this because I never left structural biology. Especially my interest was, I'm trained as a crystallographer, macromolecular crystallographer, but my interest was in the structures. So at that point of time, I developed the engineering aspects of single monomeric enzymes. What I understood is individual structures as well follow machines. Not just we are talking about ATPase, protein assemblages and natural nanomachines. Instead, even a single protein monomeric enzyme follows machines. That means it has mechanical components which are in a way flexible enough to accommodate all the stresses that go around in the microenvironment or nano environment. So same thing, I got excited with this Murburn concept. And when Dr. Murali was talking about this mucus, I have been with him for, I think, for past 4 or 5 years. We were always discussing about the ideas. The interesting thing was even in 2024, when he organized a 3-day symposium at Amrita Institute. There also I came and I was involved. Then at that time also we were discussing about all these discussions went on, and I tried to understand what are the limitations of Murburn concept, why it is not picking up like wildfire. Yes, in the morning we have heard that everything takes time and all those things. The one which was very, which I realized should be addressed is try to create a system, whether it is computational system or an experimental system, a system where we can test this Murburn concept. So again, coming back to the whole story of this, from morning onwards, we are talking about nanoscale observations and explanations or justifications, and or angstrom level. Same which I did, I finally found out in my aspartic prototype, which is at the nanoscale and angstrom level, I found out how each compartment, each machining component is assembling itself to catalyze a particular peptide or to degrade a peptide, to cut a peptide. So I never went into the chemistry part. Here also, I'm not going into the chemistry part, but I am the structural aspects I'm trying to provide to this particular theory of Murburn concept, which I think I'm very much excited about the potential and potential of this particular concept, which I'm going to talk about, that is the insertion or the So we are talking about mucus and Murburn concept and LLPS. So when he sent me the paper to look about it, I understood that what is mucus, and it has a huge role. I was also inspired, for students' sake, Men in Black. I always used to admire how they used to create these slimy skin-like structures at that, whether it was animation or really they have a manufacturing technology to create that sliminess in a particular sheets. Okay, so I was very inspired at that time, amazed about those things. So when LLPS came, this also is again related to structural biology. The structural biology aspect is I went into so depth of protein 3D structures that I felt protein 3D structures should have more flexibility than what we talk about key and what is the one? Lock and key method, sorry. So lock and key method. I never believed in this lock and key method. Exactly, it coincided with his. So when we were talking, I was feeling that yes, this particular theory is more realistic than the lock and key method. So when this lock and key method, I abandoned that thought from my mind, then what is the other one? It should be very flexible. So that led me to intrinsically disordered proteins. These intrinsically disordered proteins are responsible, and they are so sensitive that they change their shape based on the circumstances, the context of that particular. So they have multiple, they are now being called as, they can be moonlighting proteins and promiscuous proteins. Okay, there are many terminology which are coming. So the same sequence, 100% identical sequences can have different functions. So that is possible because of the intrinsic disorderliness, which enables them to adapt, manifest themselves based on the requirement. So this is nature's real design principle, which efficiently being used by nature for various manifestations. So this is another exciting part, which again aligns with Murburn concept. Okay, that spontaneity or stochastic nature which he explained. Now, the interesting fact of getting me into LLPS is I worked with the father of intrinsic disordered proteins, Dr. Peter Tompa. He's in Belgium, currently in Brussels, actually, in Belgium. So he and we worked and we published the paper. So in that, I was introduced to LLPS. So liquid-liquid phase separation. This gave me an indication that this is very important because individual molecules are not going to be, cannot explain all the phenomena that we are experiencing at the mesoscale level. So from nanoscale level, we are coming towards mesoscale level. And at this mesoscale level, we are trying to understand how individual molecules together will help in a bigger phenomena at the level of a cell level, cellular level, the sizes I'm talking about. So that aspect has I have reached that understanding that LLPS is a phenomena which is real. And when many macromolecules come together, there is a chance of crystallizing or precipitation. Aggregation happens. So interestingly, this was more interesting to me because I always look at the application part of view, not the basic science part. So this has given me an understanding that if we work on LLPS, we might even be able to try to come up with some methodologies to alleviate or to cure or to address Parkinson's disease and Alzheimer's disease. So it is the aggregation or the precipitation of the protein which is happening in the brain cells, because of which memory losses are happening or dementia is happening. So to this point, I was not aware of Murburn concept, but LLPS, then I talked to my collaborator at that point of time, and I wrote a project on LLPS, how to computationally predict LLPS, not fully knowledgeable at that time, but I wrote and submitted to EMBO scholarship, this one. Recently, about this mucus, Dr. Murali sent me the draft and told, can you add something to it? The moment I heard mucus, it is about layers of biological macromolecules assembled as they can be compartments to create compartments, they can be to create barriers and all those things. So then I went through the literature. How can we mucus and Murburn, how can we make this more physically valuable or viable through LLPS? So that's a little bit talk about it. And as you can understand, my language is very elementary because I'm just getting into this subject. I'm excited seeing the applications that are possible. Can you imagine if we are engineering LLPS with Murburn concept, we can create amniotic sacs, pregnancy related, all aspects, artificial skin and every possible thing. It has a lot of, but whatever are currently being done, their science aspect is not such as strong as what we can express or explain or justify by Murburn concept. So this is what the background is. From here, it will be almost 10 minutes that I'll close up this talk. Okay, so Murburn concept, the most important aspect is this slide, the very slide. We are talking about, we have been listening to the individual nano or angstrom level interactions which are happening, diffusible reactive species that are happening in this case. But how this particular LLPS layer, the moment we introduce the LLPS as a liquid-liquid separation, phase separation, how they are trying to control the redox reactions. So this is what the layers will be forming, and those layers are acting as not barriers, they are acting as redox regulators. So that is the beauty of this whole description. This one slide explains how this is going to happen. So here you can see this is a classical method where everything is a barrier and there are only one or two conduits which are allowing some reaction to happen, which is very difficult. However, in the case of Murburn unified concept where LLPS also is included, then we have a very a more democratic system of this redox reactions to enable various other functionalities. So classical view, I don't want to bother, but I will read it here. Classical views of mucus and redox is ROS, that is reactive oxygen species or nitrogen unstable molecules containing nitrogen that easily react with other molecules. Oh, sorry. I thought I had a mic. Okay, so this is the classical way, that is more still in a crude way. We can talk about that as look at it as a lock and key mechanism still. Okay, so the mechanistic focus on gene products, okay, and channels and membrane bond. So this is the problem with the very classical view of the reactions that are happening, trying to explain. In a way, we were hijacked, our whole idea was hijacked then. So now we are coming back here. So what are the limitations of classical mucus explanations? The one is that the explanatory gap, as in the morning also we have seen very clearly, energetic overinvestment. Just the last lecture on talk on the cyanide content, that also is about this. Okay, and there is high cost of mucus ECM and EPS systems far exceed basic protective needs. So what is that? What is the classical view? Classical view is mucus is only for defense, protection, and only these two conditions. But it is not for protection or as defense. It is actually a regulator. Okay, so that's what is being proposed in this particular paper. Okay, and this is one failure. We are unable to match the energies required to create EPS versus the protective needs. So that is one thing. The second is a failure to explain mesoscale phenomena. Exactly. So in the morning onwards, we have listened, same thing I'm telling, nanoscale level interactions we are able to. But when it comes to mesoscale, how cell is interacting with cellular receptors and membranes are interacting with individual molecules, it is not explainable by, or it is, we are unable to extend the classical theory to address this particular, answer these questions. So that is another major problem. Next is poor integration of hydrogel physics. So we understand hydrogels, as I'm telling, LLPS is there, same thing. This type of, what we have observed is proteinaceous membraneless organelles. So this is another concept which is supported by LLPS. So how these things should be explained, which are formed inside the cell, outside the cell, and they are causing a lot of diseases, many diseases, inclusion bodies, all those. So these are not being explained by our classical mucus explanations as of today. So these are the limitations, and all these things today also cannot be explained. We should be able to explain the probabilistic nature or oxidation reduction activity, and we cannot predict predictable equilibrium. So equilibriums are not justified with this explanation. So that's where Murburn model, this is now an integrative model which is able to take care of the energetics via metabolic coupling and models mesoscale complexity and gradients, incorporates stochastic dynamics and hydrogel physics. It enables us to import the research that has been done by other domains and explain that in this particular situation. To this, I need not really go through this. Already it has been covered very well. So essentials of Murburn concept is, let us talk about the probabilistic, non-deterministic in the case of metabolisms, when we want to justify how metabolisms are happening through diffusible reactive species. We are able to explain that through probabilistic and non-deterministic ways. And when it is coming to signaling, we are having a very time and space-based organization of the redox reactions. Adaptation, we have LLPS now, what we have by droplets and the inclusion bodies and things like that. Phase separation, basically we are talking. And framework treating DRS as functional. So Murburn concept is biological function emerges from stochastic aqueous redox chemistry, whereas this is chemiosmotic gradient based and membrane, and it is more rigid in its explanation. So it is not able to stand the questions, acquiring doubts of the science. Now, mucus is defined in a bit crude way as hydrated polymeric phases. And when it is hydrated polymeric phases, Murburn is able to explain this to some extent. And it has many ways of, it can be explaining the mucus layers and extracellular matrix, and we have based on charge density. These are the parameters. So let me be more honest in this case. There are models already available, which classical theories also are able to model these things using these parameters. So I listed these parameters basically for that. High hydration, amphiphilicity, and viscoelasticity, charge density. These are the major players of defining the hydrated polymeric film. Okay. So hydrated polymers are evolved mesoscale redox organizers. Regulators I told earlier, it is actually redox organizers that buffer yet exploit reactive oxygen species and their dynamics. So that is the opportunity we have by using this liquid-liquid phase separation. And why liquid-liquid? A lot of research work has gone in to are developed, and many computational models are developed, and many mechanisms have been described for liquid-liquid phase separation. What exactly is liquid-liquid? As you can see, and in the morning, Dr. Murali defined that he's looking at the cell as a colloid. Now, it has to be refined further. It is not just colloid, it is a separation of phases also. So as you can see, this is the colloid we are talking about. Heterogeneous it can be, homogeneous it can be, but there is a demixing that happens because of the inherent properties of the molecules, especially macromolecules and their properties, and ions and their properties. That is enabling this coacervate formations. So dense coacervate formations and the ions and the liquid-like dynamics. So these two are two different phases which are the output of this particular mixture. So we are not at this stage, we are at this stage. So this is the LLPS, which has been studied computationally and mathematically in a very strong. So this, when we are associating it with the explanation given for Murburn, it has become very, very strong justification, because we are providing the first physical demonstrable model for how Murburn can work and make its redox reactions happen across the cells or across systems now, not about individual molecules. So that's exactly. So intracellular condensates, we have granules, nucleoli, inclusion bodies that happen. And extracellular, we have mucus and extracellular matrix. Now, this is the LLPS and Murburn shared biophysical ground. As you can see, we have talked about these things, and this is the LLPS aspect. This is the shared biophysical background on which we can develop many experiments. And if these experiments can they be are they be restricted to only computational or theoretical? No, actually we can develop these things into real experiments as well. Okay, and I have my friend ICAR Pune, Thomas. So he developed 19-20 years back as a PhD artificial membrane. But now it is much more efficiently, we can create better enzymes or better membranes, or even forget about membraneless organelles also we can develop by engineering the concepts of LLPS with Murburn. So this aspect is about how do we explain mucus as a redox organizer in various case studies. For example, frothy mucus, how the frothy mucus in amphibian egg. This mathematically, I think they have modeled it. I just did not go into further details, but wherever my involvement was there, I was able to understand. So we have a cervical mucus, how pregnancy plugs, and ovulatory mucus, labor, all these things are very clearly now, we are able to explain based on the organization of this O2, oxygen and DRS microgradients, and exemplify that Murburn in action. These are the examples of case studies which we just submitted. It is published as a preprint in bioarchives. We are going to communicate soon this. Yeah, this is the one we are talking about. Now coming to, this is the final slide where classical failed, and using unified biophysical view, DRS dynamics are functional, enabling coherence. LLPS provides the physical architecture for redox regulation. Hydrated polymers are mesoscale redox organizers. This is the complete unified biophysical view, which strengthens the whole Murburn concept for mesoscale analysis and many, many more applications which come here. So engineered Murburn LLPS mucus analogs, we can have various models and develop engineered mucus for wound healings, wound dressings and things like that. And we can have organ on chips that can be done. And here reproductive replacements to the mucus like things. Okay, so huge. I cannot fully talk about all these things because I need to go further more in depth in this. And biofilm controls and filters, all these things are the various diagnostics and biosensors, which is my field of interest. And these are the applications. So the take-home message is very clear. Mucus, Murburn, and LLPS provides a solid bridge between explaining the phenomena and also providing a demonstrable model, physical model for us. There are many other things. So I am supposed to go a little more deeper into this. If you have any questions, okay, let me. Right.
Now we'll have my colleague, Dr. Vinith Rajathlal. He has his PhD from NIT Calicut. He is very different from all the others assembled here. He is the only guy who has come from AIML, the department I belong to, Artificial Intelligence and Machine Learning. And he's the only guy here with his full family here. His wife and daughter are sitting there. All right. So welcome, Vinith Rajathlal. We did a good work of how to use algorithmic logic to separate classical mechanism of enzyme action and the Murburn mechanism. That's what he's going to talk about. Pay attention. It'll be interesting. Thank you.
അല്ല 30 something ഉണ്ടായിരുന്നു അത് നിൽക്കേണ്ടതാ പക്ഷേ ഞാൻ അടച്ചു Direct copy presentation on the KMFRI PDF file. And Dr. K.P. Soman, who will be a dean as well, was part of this work. Amrit and Omkar, we only call him Omkar. His name is a bit long. Patange Omkareshwara Rao. They were basically our students. So fundamentally, the work is having primarily two parts to it. One is of a philosophical nature. The first part of it, I would call it a defense of Murburn from the Occam's razor perspective. The philosophy of Occam's razor is pretty known to all. We were trying to give it an algorithmic expression. So the second is again an algorithmic expression of figuring out inconsistencies in a scientific theory. So these are all basically things which are more of a fundamental philosophical nature. The second part of the work is recognition of Murzymes from its basic properties. So the second part was rather inspired, was motivated by in the sense that it could recognition of Murzymes using AI, or if the machines are themselves capable of recognizing Murzymes, that establishes that there are some fundamental nature in Murzymes which differentiates it statistically from the other molecules. So therefore, we thought recognizing of Murzymes in an automated manner would be a work towards the validation of this biological idea. So the works are basically of very fundamental nature and not as complex as the ones that have been presented here up to now. So I'll just start with so this basically, the slide differentiates the bounded reactive oxygen species, right? Because I have a problem with the biological terms. And versus diffusible reactive oxygen species. So, so fundamentally, there is a structural difference between the two. Because you might be able to appreciate it much better than I do. So my interest basically starts with this basically. The Occam's razor defense of Murburn as a scientific concept. So basically, as you know, Occam's razor is about simplicity, right? So when we say simplicity, and when we are translating that into an algorithmic framework, you need to somehow quantify the idea, right? Because machines cannot work with very abstract notions. They need something that is quantitatively expressed. So we actually, Sir came about with some measures which can probably compare two models. Okay, so we had some 8 criteria based on which we compare two models. One is the first one being shape change. So they're all, as you can see, the central column gives the range of values that they can take. So shape change that basically indicates the conformational changes that are involved in a reaction. So the greater the number of conformational changes that are involved in a reaction, that indicates a higher complexity of that. Yeah. So the second is a serial nature of the reaction. So there could be either a serial nature or a parallel nature to reactions. So serial nature essentially requires it happens in a temporal sequence, which basically entails selection of stage 1, after which there should be a selection of another stage 2 and stage 3 and so on. So there should be some fundamental intelligence that happens inside that makes the reaction proceed in a sequential manner, whereas parallel logic does not require the kind of an intelligence playing at a very microscopic level. So therefore we consider a serial reaction to be a parallel reaction to be a simpler explanation. And obviously the third is the number of steps involved. The greater the number of steps, it naturally quite obviously indicates a higher complexity for the reaction. So the third is complexity, which is again binary 0 or 1. So sir, I mean that part is complexity aspect we just explained. So so that part I think is about the molecular complexity involved in, right? So then probability is basically you have a sequence of reactions. The longer the chain of reactions, the less probable it is, assuming that at every stage you give it a certain probability, let's say 1/2 each. So 7th one is the long range electron transmission, which again is if there is a long range electron transmission, that indicates a higher level of complexity and so on. So the 8th one is the enzyme substrate complex, the structure of that complex. So so these 8 criteria were used to quantify the idea of simplicity. So what we basically did is we have 2 models, let's say model 1 and model 2, okay? And we associate a simplicity score with each of those two models. A simplicity score which is incremented every time you evaluate each of these 8 measures, okay? So a model 1, let's say for example, shape change is not there for model 1, whereas model 2 requires a conformational change. We increase the simplicity count of model 1. The same way increment the simplicity count of each of those models depending upon the values of the attributes. So finally, arriving at a measure that represents an overall simplicity score of the total model, right? So when this logic was applied to Murburn and other classical enzyme systems, we found that Murburn turned out to be representing a higher simplicity theory because it got a higher simplicity score. Second, so therefore we wrote a very simple Python code for doing this. And the second aspect of the work is the internal consistency check. So the one aspect of a scientific theory is that it should be simple enough, but flexible enough to explain the experimental observations. Right. The second aspect of a scientific theory is that admissibility from a logical perspective. Right. So it should be logically a cogent system as a sequence of arguments. So the premises or the statements of the model, the assertions of the model should not conflict each other. So therefore, that is the fundamental requirement in this regard, is that the theory should not have conflicting prepositions. So again, we listed a sequence of statements upon which consistency could be measured. So, these are the things. The first, you had something like 8 assertions. So let's take, for example, the first assertion: active site access is limited and there is no selectivity of substrates. These are 2 statements which are mutually contradictory. So if you find that in a certain mode scientific theory, these 2 things happening at the same time, occurring at the same time, it indicates that the theory is inconsistent. Similarly, again, active site access is limited, but there is no specificity of the products obtained. Then again, there is an inconsistency, and so on. So we had 8 such assertions whose presence was considered an indication of inconsistency. Okay. So again, this was used. You have 2 models. They were given an inconsistency measure. So by evaluating each of these 8 assertions, we evaluated how is there any, or the level of inconsistency that's there in a theory. So the above set of cross checks was coded into, again, a simple Python program which took 2 models to evaluate for their consistency count. So each of those 8 assertions were checked for, and out of which the total number of inconsistencies obtained was returned as a measure of its inconsistency degree. So based on that, that basically was to check, the consistency or inconsistency of a model. We found that the Murburn was largely consistent, whereas the classical models were fraught with inconsistencies. So the third aspect of it is where actually the AI comes in. That is the automated Murzyme recognition. So enzyme recognition is fundamentally identification of the catalytic nature of the compound from various features. So the general, it's not a new problem in AI, but it has been something that has been pursued by many researchers. And generally the scheme involves some standard features like amino acid composition, physicochemical properties, secondary structure of the molecule, evolutionary conservation, 3D structural descriptors, and so on. So, yeah, this is a standard pipeline for the enzyme identification. So you have an input sequence, you have pre-processing, you can extract the various features of the molecule from there, which could be amino acid composition, physicochemical properties, the sequence profile, and so on. So the third stage is rather not a mandatory one, but either you can have a similarity search from the database where you can check for other compounds which are very similar in terms of properties, or you can actually have a function prediction, which is essentially where the AI comes in. You can have an AI model which can be trained to recognize the enzymes using the features that we have extracted in stage 2. So the enzyme function annotation is to check for the result. I mean, whether it is actually rightly predicting it. Where, for example, you have enzyme commission number and so on. So which can be used to, test the AI model whether it works or not. So, and what we have found out by AI can potentially be, validated by other means. So this is the general pipeline. So automated Murzyme recognition, we investigated this problem. It's basically as an effort towards validation of Murburn as an automated recognition. As automated recognition implies the presence of distinguishable characteristics in Murzymes which machine could figure out on its own. Basically, it indicates that there is something that differentiates Murzyme statistically from the rest of the molecules. It indicates it can be interpreted as in a sense, a validation of the idea. And it also lays the foundation for a prospective tool for analysis of this class of proteins, which can aid in the discovery and recognition of hitherto unknown Murzymes. So what we did is we tried to figure out learnable discriminatory features in the structural descriptions of the proteins involved. So we basically started with protein data bank files, which contained the descriptions of the molecules. So one thing, when we started with this, we started when we passed the PDB files for the information, we started with, we thought about trying to read out the entire structure of the molecule, right? So we read all the atomic coordinates of each of the atoms in the molecule and formed a graph out of it. When we tried to process it, we found that it is, it requires a tremendous amount of computational power. So we then thought about having some other way of addressing it. So what we did is we did not read the whole structure, but we tried to read the initial portions of the parse, the initial portions of the PDB file to extract whatever relevant information that we can have. It basically involves a text process. So we read not the entire anatomy of the molecule, but rather only the description that's actually available, the textual description of the molecule from the PDB file. So the PDB files were parsed and to derive whatever structural and other information that is available from the PDB file, but then we largely treated it as a text processing problem rather than a chemical problem. We were not treating it as a chemical; we are treating it as a text. So what we did is we used a text embedding model. So we read the text from the file, fed it to a model, which generated a numerical representation of what the content, that text. So I can just show you here. So what we did is, you had the PDB file as the first stage, then it was parsed to extract some of the relevant information, whatever relevant information that is there in the text. And in the third stage, what we did is we gave it to a text embedding system. Basically, what it does is it takes text and generates a numeric vector out of it. Okay. So that vector essentially, so you can see we had a 384 dimensional vector at the middle. So this 3-dimensional vector is the representation of the molecule essentially, read from the text of the PDB file. And so that text was used to, rather train AI models to recognize Murzymes from non-Murzymes. So we used a model that is a classical model rather, because this was actually a preliminary work, a beginning kind of a work. So we used some classical models and found that, it was giving us something around nearly 85% accuracy. So they were able to recognize even without going into the very detailed structural information, some amount of differentiability could be achieved with a very classical model, a very simple classical model. So that is one, attempt at this. Second was, these are some of the statistics of the results that we obtained. So we tried various models, but SPM is the one which performed best with an accuracy of something around 84%. Right. So it was basically a fundamental work. So then the next attempt is whether we can, recognize Mursigns from structural, theoretical, and experimental features. There again, we did not go for very high-end models, AI models, but rather very simple ones, because even they were working so well. Right, they were giving nearly, with these explicitly giving the, the features of the model, we could achieve a very high level of accuracy. So therefore, we did not try any high-end computationally intensive models. So classical algorithms were largely used, and we used a collection of 20 features categorized as structural, theoretical, and experimental. So here again, you can see that the structural features are listed here. So the presence of heme, flavin, iron, and so on, you can see there. Then there are theoretical features like overall redox reaction, exergonic, oxygen needed, and so on. So there were theoretical features, and finally experimental features were used. These were given to a standards classifier, a standard simple model, which is basically a decision tree. That model is called a decision tree. So it basically, if you don't know what a decision tree is, don't worry. It's just a kind of a system which can classify cats from dogs. You can distinguish things. So so that was basically fundamentally the work. So so these are the listing, the detailed listing of the features. So, but then there was actually a problem with this because the data was very limited in size. I mean, we had hardly some 20 or 30, molecules to train the model. So there was some inconsistencies in that, or basically due to the, the lack of availability of data to fully train a model to learn the distribution fully well. So therefore, what we went on to do is that we sorted the features, the 20 features that were given to the model. They were sorted in terms of their saliency. Saliency in the sense, how important are they in distinguishing one from the other? Important in the sense that the decision tree has a measure called information gain. The higher the information gain of a feature, it indicates that it is more important in recognizing Mursigns from non-Mursigns. So we sorted the 20 features in terms of the information gain metric. Okay, and then we wrote a human logic in the sense that a rule-based system was developed based on the most important features. The values of the most important features were used to classify the Mursigns and non-Mursigns. So we use both a decision tree as well as there is a slightly more advanced version of it called a random forest. Basically, there were two things, but essentially they are all decision tree and its variants. So, so this is some of the results that we obtained, based on what we did. So we can see that the structural features alone were able to give an accuracy of something around 97%. Theoretical gave a one in the sense 100% accuracy was obtained for theoretical features, and experimental also provided nearly 100% accuracy. So these are the works. So fundamentally the works were to these are very elementary works, I should say, because we have not used any complex systems. We have not gone into it. The next level of work can probably go into the structure of the molecules to, infer, make more deeper inferences about the structure of murzymes and why they function like this and so on. Those interpretations are yet to come. So this were fundamentally some basic works done as part of, the students' works. So I was just giving you an introduction to what we started with, a couple of years back. Yeah, that's a simple application of AI into, Murburn concept. Any questions? Website are down. Website down. Actually, we had consolidated everything into a website, but then the website is not, we are not able to access the website. So I think there is some problem, some technical glitches there with that. So otherwise, we could have demonstrated how all these things were put into a web frame. Yeah, all the works were consolidated into a website, which you can access. It was actually hosted in the Amrita portal server. But now there is some glitch, we are not able to access that. Probably tomorrow or so, we could be able to correct that and make it available to.
I request Professor KMM and other speakers to occupy the seats. And since Dr. Mohan, our director from ICAR Animal Genetic Resource Institute, Karnal, is having another assignment soon. So I request him to offer his comments. Then later we'll continue our discussion. So you can occupy the seats.
Nice, Kakur.
Yeah, Mohanji, please.
Good evening to everybody. I think I can be seen and I can be also heard. Hello?
Yeah. Yes.
You can hear and you can see me. Okay. I have been hearing this Murburn concept since 2019 onwards from Dr. Manoj. And I feel the concept is now mature enough so that there are some things need to be done. First is we have to have one proof of concept. Proof of concept. So that has to be done in a isolated system. Isolated system, mitochondria, somebody was discussing as I was also there, the mitochondria need to be isolated, and in that ROS testing has to be done. Second, my thought about this is that he has described it as a stochastic process, but that is our presumption that most of the activities which take place in cell could be stochastic, but I feel it can be deterministic also, because it is our lack of understanding or that kind of a depth overall view of our thing. Our constrained view might be forcing us to tell that could be a stochastic process. Of course, it can be stochastic in one way because the large machinery inside the cell and small molecules, though they have to identify that particular location, that is there. And third thing is regarding the binding of substrates with the enzyme. Yes, that need not have to be exactly lock and key, because always it is said that the during that was originally proposed by Fisher's model. Afterwards, it has been clearly understood that as the substrate binds, the enzymatic crevice modifies itself. And also there are other things, and most importantly, there are many enzymatic processes which do not follow Michael-Menten kinetics. So not sure whether the system follows the typical Michael-Menten kinetics, because there can be allosteric effects are there. There are many things. Cooperative binding is there. There are many factors which alter the regular binding. So these are some of the things. And third thing, the practical applicability of the thing, because I was just going through how that really can be applied. There are many aspects, some sort of applied one, like especially in case of drug discovery and as well as predicting drug toxicity. This could be a very potential implications are there. And also the cancer, neurodegenerative disorders. These are very much intimated related with the mitochondrial pathology. So, is there any finding with regard to mitochondrial pathology in connection with the Murburn concept and cancer pathogenesis? So, this could be of a very great interest because mitochondria can play a very critical role in development of cancer. So that will really bring upon the practicality of the findings. So let me summarize in this way. One is the proof of concept at least in the form of isolated experiments, isolation of mitochondria and that. Second is the Michael-Menden kinetics, the kinetics of the enzymes associated with the Murburn concept. Third one is the practical value of the thing. So if we can prove, if a proof comes that Murburn concept it is having some role in the mitochondria associated pathogenesis. Not only that, mitochondrial diseases can also affect many other diseases. I'm just telling this is a simple cancer or this thing. It's not that way. There can be any disease. And last one was the practical other application that for drug testing and pharmacological utilization of this concept could also be experimented upon. I to request Dr. Murli Manoj if he is there or the presenters to respond to. And in fact, all the presentations were exceptionally well. And from Murli's presentation onwards, I'm hearing everybody's presentation. So I have been very much encouraged by this thing. This is a good thought process, and such kind of original ideas should definitely be put upon. So that's all from my side.
Hello. Yeah. Dr. Mohanan, thank you so much. The speaker, Vivian, has left because he had some emergency at his house. So I'll take your questions. I have written down all the four. Actually, with respect to mitochondria, the proof of concept is already in. The title of our paper in the Journal of Biomolecular Structure and Dynamics is Proof of Concept of Murburn Model in Mitochondrial Respiration. The proof of
that is great. That is great. That should be a very encouraging thing.
Yeah,
I'll check.
Basically, yes. So what we did is we just take ADP and phosphate and we stabilize diffusible reactive species in the form of superoxide in crown ether and dimethyl sulfoxide. Okay. And we just add it into just the solution. We get ATP. We quantified it using HPLC. So that was done in IIT Guwahati. So it's a very simple experiment, and not just there were multiple experiments done by others.
So crown either provides the crevices, that space for the experiment, that the activity to happen. Is it that way?
No, the point is our fundamental idea is it is a reactive oxygen species that couple the redox reaction with phosphorylation reaction. So that is clearly. Hello, we don't.
Yeah, I can hear you.
You can't hear me?
Yeah, I can hear you well.
Wonderful. So that proof is given by that reactive oxygen species are produced by the redox reaction, and these reactive oxygen species do the phosphorylation also. So the point that Vinith was talking about complexity, the current theory is irreducibly complex. That is the complexity. That is, you need 3 phases control: matrix control, the membrane should be controlled, and the outer phase should be controlled. And you need at least 100 odd genes. You should have 100 odd genes assembling into 4 different redox proteins, and then they should pump protein out. This is a what it's called a gambit logic. That is, first I have to invest something, then I get something back. That's not evolving. So these are all called irreducibly complex material. So we just showed that simple biomolecular reaction redox based is possible. That's what we said, proof of concept. And then all the observations in the current field is explained by the Murburn model. There is nothing that is not explained. Only what we have done is very clear, is we have completely destroyed the basic fabric, the foundation of the theories on which the classic textbook models are based. We even wrote to the Leninger authors. We asked, how is it that you say complex 5 can make ATP from ATP binding? See, the theory textbook states complex 5
Dr.
binds.
Murali, this time I would like to just quickly, can you make other observations? I understand. This is, I always enjoyed your conversation. Well, can you have some ideas on the pathogenesis, disease, and practicality like drug testing and all those things?
Yeah, so the idea of applicability and binding substrate and deterministic mechanistics that you said, the applicability and mitochondrial, just now in my talk, I don't know, you were here.
I was there.
Yeah, the Aspect Journal, American Society for Pharmacology and Experimental Pharmaceutics, they have 20 impact factor journal recent review. They said they cited 5 papers on mitochondrial therapy. They said that this Murburn model could be very relevant. And also cell stress and another paper on mitochondrial sources of cancer, mitochondrial metabolism. There were 18. The basic idea is, if we want to understand cancer and mitochondrial metabolism, there is no way skirting the issue of diffusible reactive oxygen species. And that's what Murburn does. So if you want to understand mitochondrial metabolism or cancer etiology, one has to go through the reactive oxygen species. And Murburn says that it is the foundation of metabolism. So rather than saying that this is bad, we have to say why and when it becomes.
No, it is not the question of it is bad. I tell you it the other way. If ROS plays a very key role in the disease formation, if ROS could be utilized elsewhere, so scavenging of ROS, that is the Murburn concept, can play a very major role in curation or those kind of therapeutically, it might be so important. That is what I
agree. Totally agree. And that's what I was citing. Those citations were precisely arguing for what you were saying.
Okay, thank you very much. It is really
thank you so much.
Always a pleasure to hear you.
Thank you so
no
much
doubt about it.
for your participation and support. Yes.
Yeah. I'm always. Thank you. Thanks to all speakers and thanks to Murali sir and CMFRI.
Thank you.
Thank you very much. Bye.
Anybody else? Sridhar sir, would you like to ask? Yes, please. Yes, please go ahead. He's Professor, he's Dr. Sridhar. He's an endocrinologist and he's the director of Diabetology Research Institute. He's a fellow of National Academy of Endocrinology, American, and he's also an FRCP physician. Please, sir, please go ahead.
Thank you for having me in. In fact, I belong to the ancient students who learned their biology, their biochemistry 50 years back. I've been hearing of Murburn concept forever, at least for 15 to 20 years, when you had begun publishing in Biology and Dr. Shaldakov's journals. Initially, it was very difficult to get my head around it because it challenged the very foundations on which we learned how biochemistry acts. For the sake of this workshop, I had gone through your papers, many of them, and then it began to dawn that despite all the mathematics, biophysics, and equations, the concept is fairly simple. Basically, you have put across the Murburn concept to show how energy is produced in the cell, rather than the well-established or well-documented or well-recognized phenomenon. We have shown how that is very inefficient, and your suggestion gives a much simpler and more logical way in which energy is produced.
Thank you, sir.
I
Tomorrow,
have listened to the last part of it, and I'm quite surprised that Murzymes have been classified. You use the machine learning techniques to try to differentiate the two of them. And I was also pleasantly surprised to find that you did not depend only on the AI. You added human expertise to it. So, I'm sure you're going to do this. If you're going to collect all the lectures into a manuscript, that will be a very important contribution to the Murburn concept.
thank you, sir. I hope you will join tomorrow and you will deliberate further. He was supposed to land here physically, but then this red alert psyched him out. Isn't it? You got, it's a common thing, sir, here. We have rains always. Yes. But then you are the better decider. Thank you. Hopefully you will join us tomorrow also. Thank you so much, sir. Thank you, sir.
The very fact I joined today itself shows how certain it is I'll be there tomorrow.
Thank you, sir. Thank you, sir. Yes. Now, thank you so much. Thank you so much. About the other participants, they are Afsal Manekotodi and Sukumar, sir. I think they can join tomorrow. Today it's already late. Is there any question that anybody would want to ask? And we have to have a quiz also for the younger participants. So that's the problem. So, yes. Anybody? Questions? Sir? Otherwise, if here is...
Actually, so dear students, when we were also young, this kind of lectures, it will go many times above our head. But don't worry, it will be registered somewhere and there will be some time. These are all seeds. Say, every seed will not germinate the moment it's sown. It will have its own time, its own conditions. So take it that spirit, try to learn further. And if you are not asking questions, Professor KMM may ask you the questions. So you decide which you want.
We will have questions. We have about 10 odd questions. And those who answer those questions will get a certificate of merit and also some chocolate. All right. So I think if no one has any questions, we will take down the no questions for Dr. Prasad Burra. I think it was a very interesting talk. The slides were also very interesting. So can I go on to the quiz, sir? We will start with, there are students, 7 students from first year, 5 students from second year, and 3 students from third year. Where are the 2 others? Okay. Where is Vedant? Yes. So good. Okay. So nice. Okay. So it's embarrassing a little bit. What about the part that Dr. Vinith talked about? You liked it? Yeah. So any questions? You are AI/ML people. No questions? Yes, Pranesh. Sir, he's got a question. Pranesh Subramanian. I'll come there.
In the last part, regarding the structural, theoretical, and aspect for classification, Yes. The accuracy scores and all the recall things have become exactly one. Don't you think it's overfitting the model? I mean,
one aspect of it, one possibility is overfitting because the data was too low in size. And that's basically the reason why we went the next step. Right. I mean, so relying on a model alone when the data is not so sufficient is again something that you have to think of. So then we had to basically consider the features which were relevant in terms of the contribution they gave to the classification process. And then they were sorted out, and then the biological plausibility of those features being there in the classification process was basically the human expertise that was given to it.
So that
basically was the way it was addressed. Obviously, yes. I mean, one possibility is that. The other could be that Murzymes are so distinct from the others that, it's quite natural that 100% accuracy comes.
So it has a significance for its own classification.
Because they could be they are well separated in space. Obviously, the model would be able to completely, distinguish one from the other. That could be the reason as well. It depends. Generally speaking, 100% accuracies are very unlikely. But in this case, because one possibility is they are way too different.
Okay.
Sir, I myself have a question to ask, actually. Out of all the criteria that you have considered to identify the Murzyme, which one do you think you have given more weightage or something?
Not that we give the weightage was given by the model. See, basically what it is, we have, let's say, two features that you are considering, right? The decision tree, or generally any classification algorithm, assigns a certain weightage to each of those features. That's part of the learning framework, right? So that's not something that the user gives, but rather the machine gives those weightage. Machine considers certain features to be more important than the others for the classification process.
Okay. Yeah. So, which one did you find to be, which one the machine has figured out?
It's actually there, it's in my laptop eventually. I think the heme was the feature with the highest weightage.
What one, sir?
Heme.
Heme. Yeah.
The feature with the highest weightage, which I think is biologically also.
Yeah, valid. Yeah, valid. Even I was expecting.
So the algorithm is rightly picking out the right feature.
Yeah. Even I was thinking it should be heme or maybe iron sulfur cluster.
Heme was the highest.
Maybe some flavin moiety.
Heme was the highest.
Thank you, sir.
Sukumar sir, do you want to ask something? I don't know. Is he here? Okay. So in that other kids, yes, you want some more?
I just wanted to know whether it's available. You said the website is right now down.
There was some glitch when I tried to access it yesterday.
Yeah.
It was not working, but then I think we can
back. We can correct it. Will it be, accessible from elsewhere? Everywhere? Okay. Yeah. So that would be a great help. Thank you, sir.
That's our paper in Biomedical Reviews International. So the site gives access, no money, nothing. Just go enter the details of your enzyme or enzyme system. It says whether it is a Murzyme or a Murburn system.
No.
Hello. Yeah. இப்ப ஆயிடுச்சா? ஐயோ சாரிங்க. நான் நெஸ்கி பார்த்தேன் வரல. So we'll start with the question that Vinith's talk in AML. What is the purpose of undertaking an algorithmic parsing of enzymes with AI? what is the purpose in the context of Dr. Vinith's lecture that you witnessed today? What is the context? Why did he say parsing enzymes with Murburn model or, AIML for Murzymes or Murburn system? Why is it important? What is the context? Yes, please.
So any model.
Identify yourself, son.
Myself, Harshad, third year AI student in Amrita. So why we need to algorithmically parse? Because we need to know the machine basically need to know quantitative aspect of the.
Vinith, he says machines need to know the what? Articulate.
So machines basically need to know how the protein or how enzyme works quantitatively.
Is that the purpose of undertaking the? No. Yeah, I know. You deal with these two questions, or else I can tell him. But then we all should become a part of this. So I'm afraid no, you have not articulated. Please articulate properly. Vinith.
Basically, the purpose was.
Don't give the answer. Yes, he has not yet come to the answer. Can anybody say articulate? His slide was very explicit. He started a slide on philosophy of this. Can anybody say? Use these opportunities to build confidence. You might have some kind of an idea. What will anybody think? No. Yes. Yes, Pranesh.
To basically differentiate between the non-Murzymes and the Murzymes, which are mechanistically different than the ones
right.
Like the.
No, but what is the philosophical context? The primary philosophical context. Of course, that was one. But then what will it do? The fact that if you can differentiate it with a AIML method, what will it do? What will it do?
It would eventually lead to the, we can differentiate between Murzymes from others.
No, that is fine. That you can. But what is the ultimate philosophical agenda behind it?
For further scope behind beyond.
No. Yes, right. I'm looking for what Vinith said as a reason. Yeah, you can differentiate. No problem. That is there in the question itself, what you're saying. That's not it. Anybody? So what Vinith said is, if you can differentiate it based on algorithmic AIML logic, it means there is a differentiating logic. And that fundamentally justifies the Murburn model. Do you understand? That's the logic he put there. So we were at a juncture where we had to, in spite of multiple explanations, in spite of almost 24 applications, we had to justify Murburn concept even further with that. So one logic was to have an algorithmic way of differentiating. If an algorithm differentiates, it means there is logic. So that's the grounding. Okay, nobody got that answer, right? So next question, what is the difference between sit, please? Yeah.
Sir, so said that, sorry. Sir, you said that.
Identify yourself first, son.
Train from CU Kerala.
Okay. Which
University of Kerala.
Central University of Kerala. Kasaragod.
Yes.
Wonderful.
Yeah. So I was saying that since we.
Vinith, take the question, please.
Gave the parameters on which the 10 parameters were already given, and plus the model was trained that heme is one of the examples of Murzyme. So we are already training the model.
Yes.
That this is one of the examples of Murzyme, and
Yeah.
if I am giving, is the Murzyme, if the given exam is acting like a Murzyme, that is the concept behind this?
No, the point is, he put a slide, right? One theory is, that's the next question actually. I'll ask you the next question if you can answer it. What is the difference between bros and dross?
First of all, I was confused. Why is it ROS? Why it was
Yeah,
BORS?
bound reactive oxygen species. That's the classical theory. And diffusible reactive oxygen species, that is the Murburn theory.
Yes.
So there are certain premises that agree with Bros. There are certain premises that agree with Dross. So the Bros-Dross picture shows you. The Bros is the bound reactive oxygen species, which is enzyme localized, and diffusible reactive oxygen species, which is diffusible, which can go around anywhere.
Yes.
All right. So now you have two mechanisms, and the experimental correlations, what he's talked about in that is certain experiments, certain theoretical considerations, certain structural aspects are more amenable to the Bros, or certain are to Dross. So you use, now you have a system, you give your inputs, and whatever you have observed from the protein data bank file or from your reaction system, and then you feed it, and then it will tell you whether it is agreeable more with one of these things. Okay. Make sense? Yeah. Good. Nice. So that's a good question. So you actually preempted the question, but I think his name can be written down. Today we had Pranesh. What's your name? Apurv Prem. Those people who ask sensible questions, you will get a certificate of merit, and the others will get a certificate of participation. All right. I think that's what this whole discussion forum is about. It's not just sitting idle. It's about participating. Many of the things that you learn in your textbooks, they're wrong. You're learning something wrong. All right. And here you are. You get an edge. So how do you get an edge? You don't get an edge if you don't actively participate. Don't be passive learners. Passive learning is not advocated. Curiosity, stoke it. Have a confidence. I can take on the whole world, because why? I inculcated that. Build that confidence. Ask questions, ask doubts. Feel confident to say what you feel. Pranesh did that. Who else did that here? Pranesh, Harshad did that. And then, okay. So next questions. Can I go on to the next questions? All right. 2 questions up. Then going on to the earlier question. What is the role of mucus in classical view? You know what mucus is? What's mucus?
I am Naren. Mucus acts like a protective layer for something in a classical view. In Murburn theory, it states.
Okay, that's correct. Naren, please write it. Sir, Naren. Sir, boya. Naren. Naren from our. So it's the protective view. Protective defense view. Okay, that is the classical view. What is the role of mucus in the Murburn view? Okay.
Sir, it acts as a product which can create DROS, diffusible reactive oxygen.
No, it does not. We didn't say that. Did Prasad say that it can create DROS? No, he didn't say that.
So DROS can act as something which is very important in movement concept.
Yes,
So,
but then what is the context in mucus? Okay, so you're not getting there. So who else? The models, what are the models that he said? In frog eggs, we modeled, and then also in human cervical, vaginal. Yes, who else? Yes.
I'm Ritik, second year AM.
Ritik, sir.
It acts as a regulator.
Regulator. Can you use a better word, son? Starts with M.
Modulator.
Modulator, moderator. Yes. Regulator, a little more control comes in. Something like you can have a nuclear reactor. If you put a rod in, it's called a moderator. You can call it regulator. Regulation is more. All right, good. So yes, Ritik, that's correct. It's a moderator. It's not a generator per se that way. So you see, there is something called tribochemistry. Have you heard tribochemistry? When two things meet mechanically, why does it get heated up? Because radicals are produced. From coitus to basic things, Murburn is involved. So when you catch a fish, it produces that mucus. That mechanical friction to avoid, you get friction gives grip. To avoid that, it produces mucus. So it can escape. That's an evolutionary simple cue. Secrete a few molecules, it quickly gathers water, it becomes a gel-like thing which can easily slip out of your hand. Evolutionary connection. Very simple. From there to reproductive to multiple phases. So mucus is also involved in Murburn physiology. All right, so we got the next two questions and we'll finish. Next 2-3 questions we'll finish. So give one reason why classical theory is questionable regarding oxygen. Sorry, I'll start now from the other beginning. These two latest. Sorry. What is the role of oxygen in classical textbooks that you have studied? What is the role of oxygen? Yes, son. Yes. Take son. Okay. Identify yourself, son.
My name is Naufal Naushad from Prasad.
Okay.
So in classical, it is oxygen is acting as an electron acceptor to become. It will accept like electron and convert into 2 molecules of water.
Wonderful. So his name is, he's got it right. So is it highly mobile or is it sedentary?
Sedentary.
It is very sedentary, correct. In the classical theory, oxygen doesn't move around much. It just comes and binds and sticks to complex 4 and acts like a plug. Very good. So you know what now your teachers and your professors, when you go, ask them this question. What does oxygen do? All right. If they say it just goes and sticks and stays there, this is not something that we want to, because we want to get the right thing. Why? Because so many diseases, you're aging, everything depends on this fundamental awareness. 2, what is the role of oxygen in the Murburn view? Yes. He's my student. I have already taught him. He's in the 2nd year.
My name is Pranav Sendil. I'm also from
Pranav Sendil.
AML. So the oxygen, it accepts the electrons, free electrons in the matrix and forms superoxide, which is able to...
free electrons in the matrix?
No, not free electrons, but it absorbs the electrons and forms superoxide, which is the DROs, and it basically moves through the...
What is the role of oxygen or superoxide in the Murburn view?
So basically, it facilitates movement of electrons inside and outside of the cell. So basically, maintaining the movement of the electrons, basically. So
actually, yes, you're for someone who has done my course. Okay, okay. Sit down. So oxygen is the role of oxygen is to produce diffusible reactive oxygen species, so that the electrons at one place can move to another, and oxygen is highly mobile in this view. All right, the diatomic molecule it is. Then the next question. Good, you're participating, that's good. How does cyanide kill in the classical view? How does cyanide kill in the classical view? How does cyanide kill in the classical view? How does cyanide? You want to say?
That direction.
Yeah, I'm looking, sir. First thing I did. Thank you. I like him. He said, Manoj, you should look in that direction also. First thing I did was look there, sir. Yes.
One of you should tell now that cyanide.
How is, yes. Yes, okay. He is my student. He will say, he is a PG student, my student.
I'm Ankit Devaraja. I'm from St. Joseph's University. Cytochrome in classical way binds to cytochrome oxidase, and instead of oxygen, it binds. And basically.
So what does it do after it binds to oxygen? I mean, cytochrome oxidase. So what does it do?
The binding of.
So basically, oxygen binds and sits there, right?
Yes.
Okay. Now it so it just binds and sits there. Why can't it keeps cytochrome oxidase occupied? Oxygen also does the same thing. So why is it toxic? Oxygen has no role in the classical electron. It just sits there, binds, clogging cytochrome oxidase. Why can't cyanide clog, but why can oxygen function? Why cyanide clogs, but why oxygen functions? Ha, gotcha. Yeah, sit. But yeah, okay, he participated. Please, sir, can you write down? Ankit Devraja. Anybody? See, as per the classical theory, oxygen there is an electron transport chain. Oxygen binds at last. So you have a huge pipeline. There is a clog at the end. It goes and blocks. So when water forms, it moves. So cyanide also blocks. So still it can keep on going, no? Why doesn't it? The classical theory says that oxygen, the cyanide doesn't allow oxygen to bind. But the point of oxygen binding or cyanide binding is to go and bind and clog. Affinity, cyanide, as per the classical, has more affinity for cytochrome oxidase. Huh?
Does it remove the cytochrome oxidase from the matrix?
No. Cytochrome oxidase is membrane bound.
Once it's out from the matrix.
What's that? What's that? Some other sound has come into it. Okay, I don't understand what you're saying, Ankit.
My question, what I was saying is, I felt that it was removing the cytochrome oxidase from the matrix, because once the cytochrome oxidase is away from the matrix or unbound from the matrix, the cell is...
Cytochrome, I told you, son, cytochrome oxidase is a membrane bound enzyme.
Yes.
So what is the matrix? Why are you talking about matrix?
Okay, membrane, sorry.
Huh? A membrane, okay.
Once it is coming out from the membrane.
What?
Cytochrome oxidase.
It's not coming out. It's happily sitting in the membrane. Yes. That's Suhotra. He's a graduate student.
So, where cyanide disperses oxygen, right, in complex force. So basically that disrupting the flow of electrons, which is halting the aerobic respiration, and that, according to classical theory, is halting complex fiber. Because ATP is not being produced, which is the reason cyanide is so toxic to ourselves, according to the classical view.
As per the classical view, oxygen binds and it receives electrons. Cyanide cannot receive electron. That's what you're saying.
Yes.
That's the argument. All right. That's the argument. Did you get that argument? I asked the question in a different way. So you need to be discussing with me. You can discuss amongst yourself always. The way of clear thinking, facing people, all that comes through this exercise. You chuddy buddy people, you can sit together, always you can talk. But this is where you put your confidence to test. All right. That's when 100 people can come to a consensus. 100 people can make something. But to think new, you have to think from your own shoes. And that's something that our education system does not teach. And here is one opportunity. Don't feel your teacher, he says, you should say. That's what every teacher wants. So please do it. Next, how does cyanide kill in the Murburn view? Yes, Shailesh. His name is Shailesh. Sir, I'll give. Pass it. Shailesh.
I am second year AIME student from Amrita Pai. So basically in Murburn concept, the cyanide will be binding with the DROS, which is a mediator. And
Binding.
Bind.
Cyanide binds. Okay, go ahead. Sorry.
DROS and it inhibits ATP
Binding.
clog.
Can somebody give me a better answer? Can somebody give me? Yes, go ahead. Whoever grabs the opportunity. That's it. Yes.
Actually disrupts the formation of DROS in the.
No, it doesn't disrupt the formation of.
It reacts with rocks and.
What happens?
OH is formed. Sorry.
OHR.
Water.
A water forms. So basically what cyanide does is, yes, Pranesh Subramanian said, basically what cyanide does is, my child, what cyanide does is, once the reactive oxygen species are formed, the conversion to water is so quick, very fast, that it doesn't have any lifetime. If ROS doesn't have a lifetime, all the electrical activity, catalytic activity, everything goes off. So oxygen is not needed for us. The reactive oxygen is what is needed, species is needed. That is the fundamental awareness. The fundamental way we see it is oxygen sits at one place, electrons just go and make water. It doesn't make any sense. All right. So that's the whole point of our talk. Please understand. You sat through. Okay. Give one reason why classical theory is questionable from my talk. Give one reason why classical theory is questionable of oxygen usage. Yes. Even if you don't know the answers, if you take the opportunity to speak, you are making a change to yourself. Yes, Harshad.
Traditionally, our science and classical science describes like, so they work through falsification. So you shown at the start of your talk.
I want, get my question right. And in science, it's very important that you address the question. The question asked is very simple. Give me examples where you know classical theory fails.
Sir, it just approximates the.
No. In my talk, I gave you clear-cut examples where classical theory fails.
Sir,
no discussions, please. When the questions are asked, think yourself.
Sir, RBCs lack nucleus.
Yes, RBCs lack nucleus and
Sir,
mitochondria. Yes, sit. That's enough. You got 1 point. Good. Anybody else from here? See, when I ask you, when you turn, look at your friend, smile, you're losing. You have a head. Use it. And smiling is not going to get you anywhere. Using your head is going to get you anywhere. And having the curiosity, the need to interact. Yes, please. That's good, my child.
Jashmita.
Yes. Jaswanti. Jashmita, sorry. Please, my child, go ahead.
Center. Plants lack sodium and potassium in ATPase. Yes.
You were attentive. Plants don't lack sodium and potassium. What is that exact thing that they lack? She should say. You said, what did you say now, my child? There is sodium, potassium, there is something. It's not sodium and potassium. Plants have sodium and potassium. There's no doubt about it. What is it that they lack? No. You tried, but that's not getting there. Anybody else from this side? No. Not when I ask the questions. That's when you think. That's when you think. That's not when you talk. I'm very particular about this. Why? This is what, if all the teachers who are here, please pay attention to this. When you ask a question, don't allow your students to discuss. If that happens, they're not thinking. They're just dissipating. You need to think within yourselves and you need to be coming out with something. That's when you have a thought process. Damn it. That's very important. If you're not doing that, start doing that. That's what you take home from this discussion forum. That's what science is about. You start thinking for yourself. All right, my child, don't get upset with me for that. You are pursuing science. Science is that. Otherwise, you go kindergarten, baba, black sheep, evil. You can say it, whatever they say, you parrot. Right? We are not doing that. Good. What is the other example that I said where you can challenge?
It shows whether are you attentive. You want to say something? Take the opportunity, my child. We are all staying back. You know, it's late. I know. Yes. Yes, son.
Thermodynamic feasibility.
Yes, that is there. But I think we can write that. Yes, those are specific, instances. In one slide, I put 3 figures. Remember, one was about erythrocytes lacking nucleus and mitochondria. Yet it survives 120 days. And then Vasudevan sir said, oh, it was something that was even, perplexing me when I was a student. At your age, when I studied these things, I thought it was not possible. Logan, those are mechanistic aspects. I gave specific examples where the classical system fails, which you see on a regular basis. If you're a student of biology, you should question. So when you see something very regularly, it doesn't appeal to you, but yet you go over it, you're losing opportunities. You're becoming a parroting mechanism rather than a thinking person. That's what I want you to think. Okay, these are examples. Good, you are paying attention. That's nice. Please, sir, you could repair on that. What's your name, sir? Naufal. Very good, Naufal. Then, other 2 examples. No, don't discuss. Don't discuss. If you discuss, 2 brains they come together to work like a team. 2 single. Yes, Harshit. Hartik.
Cyanobacteria.
Why cyanobacteria? Yeah, even other bacteria also are aprotic. Why cyanobacteria particularly? Yes, many bacteria are also dimensionally small. Why did I particularly say cyanobacteria? Cyanobacteria is right, but you're getting half right. What is the rationale I said cyanobacteria? Yes, Shailesh?
Photosynthesis and respiration.
Correct. Why photosynthesis and respiration is incompatible in cyanobacteria?
Needs water, doesn't need.
Wonderful. One makes water, another breaks water. When you have an electronic circuitry, with common element, it cannot work both directions. It will not work both directions. Only the thermodynamically downhill one will work. The uphill one won't work. Which one is the uphill one in those 2? Respiration or photosynthesis?
Respiration.
Which one is uphill in photosynthesis or respiration? Respiration, all cells do. Photosynthesis, only some cells do. So which one is uphill? Come on. Photosynthesis. Okay, thank you. Sit. Shailesh and Hrithik, half. Give one. And then what was that plant cell? Remember? Plant cells do not have sodium-potassium pump. Sodium-potassium pump. They have sodium and potassium. Hearing is not good enough. There is a protein that is supposed to pump sodium out and potassium in. Jens Kuh got a Nobel for that. That's what is supposed to give all our nerve activities, keep sodium-potassium imbalance, and all that is supposed to work for us. So now plants don't have it, that protein. Yet plants have high amount of potassium. That's one classic example. Okay. So last question of today is, give one reason why classical theory of cyanide toxicity can be challenged. Why did Vivian say that classical theory of cyanide toxicity can be challenged? Why do you think the classical binding based affinity based theory was wrong? Yes, Suhotra says
there are more.
One reason. There are multiple reasons. Give me one reason.
There are more cytochrome C oxidase binding sites and the molecules of cyanide itself.
Yes.
I don't know the exact number, but there are not enough cyanide molecules to go and bind to
Correct.
individuals.
Correct. That's a correct answer. One is the dosage. You have very small amount of cyanide acting. You need oxygen only once in 2 minutes. You take cyanide, immediate knockout. Carbon monoxide, which is a much stronger agent, million times more stronger binding agent than cyanide. It takes 40% to get into the blood for you to get lose consciousness. Cyanide, you have 2.5 to 3%, you're done. There is no return ticket. All right. So why is a binding based rationale, stronger binder, not able to kill even at 40%, whereas a weaker binding agent is able to kill? So cyanide, that's one dosage, cyanide dosage. Then second, any idea? KD value, that's a binding based argument. Then KD, yeah, that's a binding based argument. There was another argument quantitatively. He said KD is KM. I don't think you will know. Okay. KD is equal to KM plus something. In the experimental system, they found that KM is in micromolar, whereas KD is in millimolar. Mathematically, as per the classical theory, it is infeasible. That's not admissible. Correct. But then you did not articulate it properly. Okay. So kinetics, what was the kinetics part? Classical, why can't it classical theory explain kinetics? All right, no problem. Classical theory, if it were to binding based experiment, if you do, the binding based is too slow, it will take a longer time, but cyanide knocks out in seconds, right? Kills you. All right, thank you so much. That's good. So today's, we have got some written names. These people will get certificate of merit, other will get certificate of participation. That's just a way for us to tell you that when you sit here, you have come so far. The fact that you have come here, why do you come here? You are getting latest, unpublished things that are not in your textbooks here. What can you do with it? It should inspire you to think different. All right. Otherwise, there is a lot of information already out there. And here you get to interact with people who are not familiar with you. You can discuss science. That's what a discussion forum is about. So when a speaker comes there tomorrow and all of you sit, your teacher, make your teachers, proud by interacting, participating, being present. Okay. Thank you so much for your attendance. And hopefully we'll see you in greater numbers tomorrow. Today we had rain, but yet you people came around. Thank you so much. Wonderful, wonderful, wonderful. All right. Does anybody have anything to say? We can go in 2-3 minutes. Does anybody have anything to say? You can say. There's nothing asked now. Use this opportunity, kiddos. Feel confident. Sir, everything parayu. Tra dure mannalle. Valanjeri num. Avadnum. Harshad, you want to say?
So in one of your slides, you mentioned Murzone, right? So I just need a clarification about it. So does Murzone is entitlement of the zone where those reactions, the DROs, all those things happen? So or else I had another doubt, like, is it like you are just confining the space where it happened?
Like you have an orbital, which is a probability distribution of finding an electron. Murzone is a greater probability of finding a diffusible reactive species. So if you have a heme protein, there's a good chance that you will find a reactive oxygen species nearby. That's it. That's it. That's a Murzone. Okay. Yeah.
Yes. Tell something.
My name is Maya. I am assistant professor from MES KVM College. This was a good session. It is we have already know the chemiosmotic model, but it's new concept for me. But thank you, sir. I want to study more, sir.
Okay. I like that. I like that answer. That. Yeah. Use this opportunity. don't get anything with this. But the fact that you have come here scientific, don't look at each other and smile. Yes. She is my student's mother. Rita's mother, first year student's mother. Yes, come.
Hello.
Learning never stops. Wonderful. Madam, please.
My name is Saranya. After listening everything, I came to a conclusion. Our elders used to say ozone. Early morning you go walk. Ozone is also a reactive species, oxygen reactive species. So when we inhale, these are all the benefits we may get. We may generate more ATP with that ozone.
That I don't know. I don't know.
Like
that is, I don't know. I don't know. The point is, we know for sure when we have, there is IR therapy. You know that? Photodynamic therapy. Some of the people were supposed to join today. There's Danny Aiello. He photodynamic therapy. He is a chiropractor in US. He wrote a book in which he discusses Murburn concept extensively. So in that, when you use red light, the reactive oxygen species is formed. You get more ATP. All that is known. It's experimentally proven. So I don't know about ozone generating more ATP. I don't know. As far as I'm concerned, I have singlet oxygen can also. Ozone I have really not gone into, but there's a possibility. I don't know.
And also, I heard a thing that when pneumonia patients are exposed to sunlight, they are recovering soon. So this may be the reason.
Could be, could be. There are so many photodynamic events. Yes, ozone is Ross. Definitely. That I know, but does ozone give ATP? That I don't. That is what. See, he just cross-verified and he was telling. Wonderful. Wonderful, wonderful. So yes, see, I wanted to tell you something. There are so many medical practices that will be streamlined in future. Acupuncture, homeopathy, there are so many things, practices in them, which we can rationalize now. Earlier, they used to be treated, suppose you touch here, your pain here goes. There is reproducibility, some reproducibility. But it cannot be explained by classical theory. Now we can try to approach. And in say classical, traditional, not just in our Ayurveda, in Chinese, in South American traditional medicines, there are hot food. All right, some food, adu choodu ananu. There was no basis for that in the modern. Now, but we know small amounts of chemicals, if they go, they can lead to uncoupling, coat uncoupling, and it can generate a greater amount of heat. So we know the rationale for a generation of heat. It's interfacial Ross modulation. So that's another thing. So many things will be now better sensible to us. Thank you so much. Thank you, madam. Thank you for the participation. Yes.
Thank you,
sir. Sure. Please. Anybody is free to talk. Huh? Yes.
Yes, I am Dr. Jojo Paul. I was a professor in commerce. I'm a layman to this. I retired 10 years back. Why I am interested in it? I am a Marma therapist also. I do healing to others. So out of curiosity, I came here to listen to this.
Wonderful, sir.
Okay, my suggestion is this. I don't have any doubt. Why? Because I am a layman to this. I'm not a technical or medical person.
What healer are you?
Marma therapy. Marma. Varma.
Oh, okay,
In Tamil Nadu, it is called Varma. I was into holistic healing system, now turned into this Varma therapy.
Okay.
I used to give healing to others also.
Yes.
So my suggestion, humble suggestion is this wonderful contribution to the science. Can you make it layman friendly, a literature or not textbook actually, a literature, a handout, which could be used as a promotional material or instrumental to the conscientization program,
Sure.
awareness creation
Sure, sir.
program.
Sure, sir.
We used to arrange some camps also, medical camps. And actually, I'm very proud to say that I am involved in the public health conscientization programs also. So if we get some handouts or something like that in a very vernacular language or in a layman's language, it will be very useful.
Thank you, sir.
I
It's taken.
thank you.
Actually, Menon sir has got a journal called Shastra, which is released by Swadeshi Science Movement Kerala. And I had actually written as simple as possible for me, but we will try to make it even more, accessible and approachable.
That English language, I can give. Yeah.
Yeah. Yes, sir. Yes, sir. I understand totally.
In English, that
Yes.
I can.
Yeah. We did that. The sir had generated this. This is a very legitimate suggestion.
Just get them know.
Yes, definitely. Anybody else wants to say something? Yes, Febin. Febin is my student from Mala in 2007.
2006 to 8.
Yeah.
2006 to
Yeah,
8.
yeah.
Very interesting lecture, sir. One question for you is, like we know about antioxidants.
Yes.
There was one mention
Yes.
in your one of the slides.
Yes.
So the purpose of antioxidant is to actually inhibit the response of superoxide or
Yes.
reactive oxygen species.
Yes.
So our system already having antioxidants, inbuilt antioxidants are there.
And antioxidant enzymes also.
Enzymes also there. And proportionately now in medical science, we additionally give the patients or like in supplement industry, we additionally give the antioxidant in additional amount. For example, in Corona during
Correct.
time,
Correct.
people used to give supplement with vitamin C or
Correct.
like lot of things.
Correct.
So like in the model, what you mentioned, like reactive oxygen species are very important for propelling the reaction further. So
It is important for all of your physiology.
yeah.
Yes, go ahead.
So we are debating that it will be helpful for moving the reaction forward. And in another side, in our system itself having antioxidant, and if you are giving in a supplement form additional antioxidant, it do not suppress the ROS, like how the body will be reacting
That's a
towards.
very good question. The whole world is going for antioxidants. They did research. You give too much antioxidants, it is counterproductive. All right. So the point that I stress there is in what amount, in what place, at what time. These three things determine whether it is good or bad. So when I was working in US, I don't know if you recollected, I said that my job was to minimize the ROS during drug lead optimization. So I said, if you take away the ROS, the reaction doesn't. What happened is there is some enzyme called superoxide dismutase. So they used to add this superoxide dismutase to eat away the superoxide. So they thought that superoxide is not needed. But then what I did is, they say vitamin C, it is a superoxide scavenger. So I put vitamin C, nothing happens. But I put a fatty acyl substitution. Just move the raw scavenger to the membrane area, poof, the reaction stops. So where it produces, how much it produces, and at what time it produces, that is what's in. So the diffusible reactive species production is sustained release dynamics. There is not bulk production. That bulk production happens in a very uncontrolled way during cancer states and all those neurodegeneration. That's when it goes awry. Research is already there. It gives inorhabditis and all that antioxidants. It doesn't grow properly. So oxidative stress, which Helmut Sees and other people used, oxygen was seen primarily giving rise to this reactive oxygen species was seen as a toxic wasteful product. Oxygen in itself, zero value. Oxygen becomes diffusible reactive species. You have electrical activity, mechanical activity, thermal activity, chemical activity. That's why you need oxygen. ATP is just a part of the picture. ATP is just a part. I'm not going to undermine ATP because I've explained ATP formation. We have explained ATP formation. But ATP Currency, you take currency. What and all can you do? Money can't buy you. There are some other things also. Just like in society, ATP is seen as an arranged currency. Now in society, can money buy you everything? No. There are human relations. So why is Murali sir sitting here? Why is Menon sir sitting here? They are not sitting here for money or anything. There is some other purpose. So similarly, certain other things get done in cell, not just by ATP. We have to explore all that, so many things. All right. Anything else? Otherwise, we'll want. Yes, Rishikesh. Huh?
Sir,
You can say. It's free for all. Sir, what are you?
I had a question for you.
Yes.
Sir, if the mitochondrial complex 1, if isolated and buffered at pH 7.4, and if the ADP is maintained, can we, obtain ATP if ROS is applied? And if that is possible, which type?
Sorry, can you repeat? In the middle, our convener said something, I lost track.
All right, I'll repeat it, sir. So I'm asking whether if mitochondrial complex 1, if isolated and buffered at pH 7.4 and ADP is maintained, can we obtain ATP if ROS is applied? And which type of ROS catalyzes ADP phosphorylation?
Oh, yeah, wonderful. I actually said that Kathleen Naylor had actually done an experiment where she took in 1990, 80s end, she took mitochondrial same buffered system and she gave only xanthine and xanthine oxidase. Xanthine and xanthine oxidase generate superoxide. It's known. ATP production goes up.
Okay.
So clearly you don't need any gradient. Yeah. Clearly you don't need anything other than ROS. Not just that, another European group took the same thing in erythrocytes. In erythrocytes also, ATP production goes up. So superoxide, when it is present, it can generate peroxide, hydroxyl radical, all that. So which one? The theoretical treatments show superoxide is capable, hydroxyl radical is capable, all that. So feasibility is there, lot of feasibility. All right.
Okay, my second question is, you talked about the ROS and its role in ATP production in the cell.
That's so part of the picture.
Yes. And so we all know ROS is pretty, not good for the cell in high quantity. So if you mention that inside the cell, if ROS is there, the ATP can be maintained. Extreme amounts of ROS can also damage the cell, right?
It does.
So what can you say about that? Like,
yeah, that's what I'm saying. I said clearly that anything in right amount, at the right space, at the right time is good.
Yeah.
You like glucose, don't put it in a place that it is not supposed to be. It will be a toxin. Same is with ROS. I just said that. All right. So ROS at the wrong place, wrong amount, wrong time is bad. So is every other molecule. Every other molecule in your system, wrong place, wrong time, wrong amount, bad. So is ROS. I say ROS, I don't say always ROS, I say diffusible reactive species. Because in anaerobic system, you don't have oxygen-centered diffusible reactive species. So I want to encompass the whole. You imagine something happens here. How does the cell know here? The whole cell react. The only way is the electronic connection through the diffusible species. It's obvious, it is imperative that diffusible reactive species restructure things, communicate things, electron transfer. The only force that works at our level, at the physics that life is electromagnetic force. So oxygen works with that. He corrected me saying that you should use electrostatic. It's okay, same for me. Physics, yeah, we'll wind up. Kiddos, there is tea and snack outside. Thank you so much. Thank you so much, my dear folks. Tomorrow, 9 o'clock, please be here at 8:45. All right, please be here so that we'll start in time. We lost so many people here today, because of rain. And tomorrow we will not have rain, hopefully. And most of you are here. We want more interaction. Okay? Okay? Please have interaction. Come with that zeal to interact. This is a discussion forum. The maximum participants are you people, kids. You know, you're young budding scientists. Yes or no? Huh? Yes. Please interact. Thank you. Thank you so much. Be nice. Have a good night's sleep. All right. Bye. Yes.